Hello everyone, welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, I'm Associate Professor in the Department of Chemistry and Biochemistry at Florida State University. I do light matter interactions, color chemistry, dyes, pigments, but more importantly, joining me today is Dr. Angela Knapp. Angie, if you wouldn't mind telling the audience about yourself.
Sure, so I'm Angie. I study the marine nitrogen cycle. And one of the reasons we care about that is because it limits phytoplankton growth both on land and in the ocean. And just like if you go to Home Depot and get a bag of fertilizer and it says NPK on it, plants in your garden need those nutrients, plants in the ocean need those nutrients too. And we think that on glacial, interglacial timescales, so on timescales of 10,000 to 100,000 to a million years, that plant growth in the ocean has played an important role regulating atmospheric CO2 concentrations. But we also think in the modern ocean that over vast regions of the ocean, plant growth is limited by nitrogen availability. So if you care about understanding what regulates atmospheric CO2 concentrations, and you think that plant growth in the ocean is important, but that plant growth is limited by nitrogen availability, you start to care about what the sources of nitrogen are to the ocean, as well as the processes that remove them. And so that's what we study in my lab. And nitrogen is actually sort of like the Goldilocks element for phytoplankton, because in truth, other elements like phosphorus and iron limit their growth as well. Those have strictly physical sources, so the weathering of rocks on land and delivery of those elements to the ocean by rivers. Nitrogen is different. All the inputs and outputs are biologically mediated, which makes it really interesting, but also really complicated. So we have to understand biology, geology, chemistry, oceanography, and I'm a geochemist, so I use stable isotopes as a geochemical tool to trace the sources and sinks of nitrogen to and from the ocean. And so, yeah, that's what I do. And my work is complementary to a lot of, I work with a lot of biologists as well to try and understand the microbes that are cycling nitrogen to and from the ocean. So that's what goes on in my lab.
Sounds good. Arguably equally as important, what game are you playing tonight?
Right. Super Mario World. Yeah, classic.
All right. There it is, everyone. Welcome to Ask a Scientist Gaming. We have Angela Knapp playing Super Mario World. We'll let you get started a little bit before bombarding you with questions. So
all right.
Go ahead and let's dive in.
Okay, here we go. This is my
How long has it been since you played this game? Prior to this week?
Yeah, prior to practicing with my kids. It has been, I think I first started playing like when I was probably in second or third grade, and I had friends who had it, not me. And so I just remember hanging out in their basement and playing this game with them.
That's awesome.
Yeah, I think there were some slumber parties, and I think there was Gem and the Holograms involved sometimes. I think they watched some of that. And
temporally appropriate, yeah.
And played a little Mario, So, yeah, one of those classic suburban childhoods. You're going to find, so this is all dependent on muscle memory at this point, and
that's what makes it fun. All right, so you told us a lot about, research and whatnot. How about you and the path that got you here?
Sure. So I grew up in the Seattle area, suburbs, and so let's see, I did my undergraduate at a small liberal arts school in Washington State, Whitman College, where I was a biology and geology major. Football players can get me down. And a small liberal arts school is great, but they don't have the breadth of a school like FSU. And so I knew that I really liked the intersection of biology and geology. I also took some chemistry. I took full semesters of organic. But I didn't really have the verbiage to figure out how you would combine those. So, so I ended up taking a year off after undergrad, and I worked in a chemical oceanography lab. at the University of Washington. And that's where I sort of realized that oceanography was actually sort of a really good combination of biology, geology, chemistry, all the parts that I really liked. And it had, I knew I was interested in trying to understand what controlled climate change. And so that seemed like it was really sort of the perfect intersection of all those things. And so from there, after I took a year off and worked as a tech at the University of Washington, I did my PhD at Princeton. And then I did a postdoc at the University of Southern California. You guys, I'm never going to get Yoshi. I did a NOAA Climate and Global Change postdoctoral fellowship at the University of Southern California. And then I went to the University of Miami and was a postdoc and research faculty. And then I ended up here. So I've been to all four corners, and my husband is from the middle. He's also a scientist, and we have two girls along the way. So we have a 12 and a 7-year-old daughter. So that keeps us out of trouble.
I did not know you were a postdoc at USC. That's where I went to grad school. I was there 2005 to 2010. Yeah.
Yes, so we overlapped. Because I was there 2006 to 2008 in
That's
the
awesome.
Environmental Biology department.
Small world. So you did the, what is that? The Wrigley, what's the island that has the...
Yeah, I never made it out to Wrigley. It's
Oh, wow.
ridiculous.
Yeah, because they
know.
have...
How dumb is that?
Like dedicated. So for those of you that don't know, USC, there's an island. What is the name of the island? I can't remember it. Catalina Island. And so it's like this tourist trap, but USC owns a little marine research area in there. And so we'd have like tiny conferences there once in a while. So I've been there 3 or 4 times, and you haven't.
Yeah. No, it's really silly, actually. Didn't make it out to Wrigley, which is a shame.
There's still time.
There is still time. That's the right attitude. Exactly.
Yeah. As soon as COVID is behind us, everyone's on their way.
Yeah. Everybody's going everywhere. Yeah. So we did overlap. I don't remember where the chemistry building is there.
There's like 3 different ones, but
yeah.
All right. Quick question before I open for questions. What are we drinking tonight?
I have it's a gin and tonic. Yeah, that's what I have. It's not quite the classic one. I used a mixer and I'm out of line, but I'm improvising, like everybody in COVID who doesn't quite have exactly what they want. So.
Yeah, so what's going to be fun? So I'm drinking a Blue Moon right now, but my plan when we do this in person is I'm going to drink whatever my guest does. Just
nice.
diversify my experience. And Skuzbot Wotus just redeemed Take a Drink. So pause, let's take a drink. Cheers to everyone joining us on a Wednesday night and drinking. Here's to science and video gaming.
There we go.
All right, let's open the chat for questions. Angie is ready to answer anything you have. We'll start with a fun one from Triboluminescence. What's your favorite nitrogen source?
Oh, easy. And I could talk about this all night. I'm going to kill myself playing it in the meantime in Mario World. Trichodesmium is easily my favorite nitrogen fixer. So it is a cyanobacterial diazotrope. Was this a plant? Is somebody out there know me? Just trying to get me to talk. I love it.
I think I know who that is, actually, but
okay.
So
Trichodesmium is this cyanobacterial diazotrope. It was actually observed by Darwin. Everybody who lives around the coast of Florida and goes to the beaches here has probably seen it. So it looks like sawdust. So it forms these blooms, massive blooms that can be seen from space. It's amazing. It breaks all the rules. But it's quantitatively one of the most important nitrogen fixers in the ocean. It's the biggest. I could talk about it forever. I could spend all night talking about it.
Chelsea Brockdock says that was a predictable answer from you. So they definitely know
Chelsea?
you.
Okay. Hi, Chelsea. Thanks for coming. I'm like, somebody knows me.
That's awesome.
Yes.
Wait, aren't volcanoes a nitrogen source?
A very small nitrogen source. Yeah, so they do, when volcanoes erupt, they are so hot that they actually fix atmospheric N2 into nitrogen oxides. And some of those fall into the ocean. And that can stimulate phytoplankton growth. So this is like, I'm getting a bunch of planet questions here. This is awesome. You guys are the best. I don't know how else out there.
Yeah, keep them coming.
Yeah. So in 2000, I'm going to lose Yoshi. It's okay. In 2018, the Kilauea volcano in Hawaii erupted, and they noticed, so it erupted in April, and when lava hit the ocean in June, within a matter of days, it triggered a phytoplankton bloom, and it started to extend away from the site of where lava entered the ocean. And there's a really, really great oceanography department at the University of Hawaii, actually run by an FSU alum, Dave Carl, got his master's here in oceanography back in the day, and he's a National Academy member. And they got a boat and they went out and they collected samples. And Dave Carl actually thought that it was Trichodesmium. It's his favorite. I bet it's his favorite phytoplankton and nitrogen fixer too. It's like everybody in, yeah, it's easy to love Trichodesmium. But
somebody clip that, please. That's quoted forever. That's amazing.
Truly, it really, so they thought that it was going to, so one of the things about Trichodesmium is that it's often limited by iron availability. And when you've got all this lava erupting, it's full of iron, it's basalt, an iron-rich mineral and rock, or there are other minerals that are iron-rich. Basalt is not a mineral, but the rock has a bunch of iron in it. And so they really thought that it was going to be stimulating Trichodesmium, and that's what was going to be blooming. But they went out there, and there were molecular ecologists who sequenced the DNA of the phytoplankton, and it was not Trichodesmium. In fact, there weren't any nitrogen fixers out there. And so what they instead saw were some diatoms, which is interesting because diatoms usually live in coastal systems where you have upwelling, and they usually eat nitrate. And so they were like, what the heck is going on? Oh, sad. So they wanted to know why the diatoms were going, but they noticed that there was actually high concentrations of nitrate in the surface waters in this strip of water where the phytoplankton were blooming that was impacted by the Kilauea volcano. And that's unusual. We think of the waters around Hawaii as an ocean desert, and there usually is not nitrate in those surface waters. Guys. And so they collected some samples and they knew that I measured the isotopic composition of nitrate, and they sent it to me to ask me to try and identify where it came from. And it was really kind of a mystery for a long time, but it was clear that this nitrate that was associated with the lava from the volcano entering the ocean was driving the phytoplankton bloom. And what we learned is that, God, I know. But I don't have Yoshi, like I'm stuck. So, and I don't know where all my bricks are, but it's so sad, you know. It's okay, guys. You're going to notice that I can't talk and play video games at the same time. So I'm going to try and get the science right, and then...
That's fine. So we got a request in. Oh, sorry. Go ahead and finish.
Yeah. The nitrate came from upwelling. So I can talk more about that later. But yeah, so the volcano, the lava flowed downhill, and
that's awesome. That's a relatively recent discovery. That's something we didn't know prior to that. That's amazing. All right, Reese's Pieces has requested a factoid. Do you have a mind-blowing factoid you want to drop on chat?
Let's see. Ken, help me out. I fed you some.
I don't have them in front of me.
Oh, no, I don't either. Oh, shoot, dude. I could go, my computer's over there. I could go find them. What are my factoids?
Let me see, you sent me some. I can look them up. In the meantime, we will come back to the factoid.
Okay, I'm sorry.
One of the general questions from Bronco is, how do aquatic plants use nitrogen?
Right, good question. So mostly they use it to build biomass. So to build proteins and DNA and RNA, enzymes, all those molecules. So all those really fundamental biomolecules that you and I share as well require a lot of nitrogen. And so after carbon and hydrogen and oxygen, nitrogen is one of the most important components of our biomass and of all living biomass. So non-negotiable, must have it for DNA and enzymes and proteins. Yeah.
Sorry, I'm trying to look up factoids right now.
I know. Sorry. Should printed out my list. It's in addition to not being able to catch Yoshi.
You have one on glacial and interglacial cycles over the past 1 million years.
Okay. So, yeah, I guess the factoid is, how do we know that phytoplankton in the ocean are responsible for atmospheric CO2 drawdown on glacial and interglacial timescales? And there are two parts to the answer. And really, we care about nitrogen mostly because it's in the service of carbon, to be honest. I'm going to die. Oh, no. But how do I can't. No, it's not going to work. So let's see. Glacial and interglacial timescales. So phytoplankton growing in, I'm going to have to just, carbon, atmospheric CO2 concentrations that increase and decrease on the timescales of 10,000 to a million years have to be driven by changes in the ocean because of the amount of carbon in the ocean compared to other reservoirs of carbon on the planet, like in rocks on land. That's where most of the carbon on the planet is in rocks. We're never going to pass this level, guys.
You should go do the yellow switch. Go the other way and get the yellow switch.
That's right. That's why I always do that. I just forgot. I'm like, why do I always go left first? I'm going to mix it up and go right.
There we go. Bronco, thank you for the tier 1 sub, 3 months. If you didn't notice, we now have 2 emotes for tier 1 subscriptions. Thanks to the generous donation of a bunch of people as well as gift subs, we have now 2 tier 1 subs or 2 emotes for tier 1 subs. So you get the alcohol and the rage one. So pop those up on the screen. So enjoy. Again, thank you for supporting the channel. We don't need the financial support. We appreciate it. When people are here in person, it'll pay for alcohol and Uber rides. Right now it pays for gas to drop off green screens. So
you're not kidding.
Yep.
So it's not a good factoid. It's a long story. But basically, the amount of carbon in the ocean and its residence time indicate that it's the ocean, man, that controls atmospheric CO2 changes on glacial and interglacial timescales, and not plants on land, and not.
So it turns out the ocean is important.
Turns out. If you care about carbon cycling on longer time scales, for some of the people who are here, then you care about plate tectonics and carbon cycling through rocks. And I know it seems like we have some of those friends here today, so we got to give them a shout out.
Sounds good. All right, so we have another question. I'm going to catch up. Going back, Chelsea wants to know, are microbes more likely to require specific metal cofactor for enzymatic processes or be able to substitute in other metals? So first translate that question and then answer it.
Okay. So, dear Chelsea, I love Chelsea so much. She's one of the best graduate students we've had. She just graduated, is off to a postdoc somewhere. She's fabulous. She TA'd for me.
Shout out to Chelsea.
Yes, Chelsea's lovely. So good question, Chelsea. It depends on the enzyme. I'm never going to be able to play this and talk.
Wait, let me translate this question first. So biological things have metal centers like, zinc and iron and copper and all sorts of different metals that do process chemistry. They do reactions. They facilitate those reactions. But sometimes those metals aren't available. So the question is, I guess, I don't know how you want to translate the question from that.
So what do they do when they can't get what they want, right? And they have different strategies. So sometimes they substitute another metal in, and sometimes they substitute with other things. And that is not my expertise. So I am not even going to go out on a limb and say the wrong thing. I think it's enzyme specific. So for nitrogen fixation, there are different. Yeah, I don't think they substitute. It's like you have iron or you don't, and that's it. But there are some enzymes can substitute in other elements. There are other molecules like phospholipids. We have learned, some colleagues up at Woods Hole have discovered that phytoplankton and microbes are really good at substituting phosphorus out of lipids when phosphorus is scarce, and they can make lipids out of sulfur and nitrogen, which is amazing. So the lipids are in our skin, the cell membranes and things like that. So all living things have cell membranes, and there are lipids in there, and usually we think of them as being phospholipids, so phosphorus being an important constituent of those lipids. But marine microbes play tricks. There are a lot of places where phosphorus runs out, and they substitute other things, so they get resourceful.
To quote Ian Malcolm from Jurassic Park, life finds a way. It always finds a way. There's examples of new organisms or derivative organisms in the Chernobyl fallout area that harvest nuclear waste. It's crazy.
God, all right.
And there's bacteria that eat bacteria. I don't know if it's bacteria, but things that eat plastics, and plastics didn't exist prior to the 1950s. So we've created things that they have eaten since then, which is just insane.
Right. Microbes will figure it out. Although it's interesting because, the most abundant enzyme on the planet, right, is rubisco, so the enzyme that carries out photosynthesis. And we actually think that's one of the most conserved enzymes around, right? And it hasn't, and same with the enzyme that carries out nitrogen fixation. We actually think there's some mathematical modeling that suggests basically some organism in the community has to take it for the team. It's this huge set of genes, and it's really unwieldy. And so they're it's I think the analogy is the Black Queen hypothesis, right? So it's the analogy is the game of hearts, where somebody has to have it, and somebody's going to get stuck holding the hand with the really onerous high obligation set of genes. But Yoshi, sorry.
Yoshi's revenge.
That's right.
Sorry.
I'm taking you with me, lady.
Poor Yoshi, betrayed.
No, I do love Yoshi. So I don't know.
All right.
So some enzymes can substitute. Others, it seems no, they don't substitute. They don't want to.
I mean, that's crazy how subtle and intricate the body is, and our biological things are in terms of processing, and yet on the contrast, they're so robust that they survive anything. It's a weird dichotomy. Yeah.
It's I'm trying to figure out, which your pet, not critter, but microbe is. So it's interesting.
All right. So Bronco wants to know, why is the ocean blue? So that's actually an optics question.
Go for it, Ken.
Do you want me to answer that one?
Please, my husband's good at this one.
It's basically for the same reason the sky is blue. And so it's because of scattering light. And so the light off the sun is white. I mean, generally white. I mean, it has certain dominating features, but we'll say it's white light. And so what ends up happening is light gets scattered. The shorter the wavelength of light, the more it scatters. And so higher energy blue light scatters more than orange and red light. And so what you have is essentially the red and the orange and whatnot go deeper into the water, but the blue light scatters on the top. And so it gives an appearance of blue. So, oof, that was close.
Thank you. See, and I got further in Mario now.
That's yes. Scuzzbot just quoted, it's Rayleigh scattering. And so it's size dependent scattering. So it's the interaction
you guys
wavelengths.
are the best.
So,
all right.
Yeah. All right.
Go team.
Yeah, exactly. This is a team effort. So Ninja Physics. I can't help but ask if you've seen Seaspiracy on Netflix.
No, I have not.
It's been out for like 2 days.
For it. There's not a lot of TV watching in my house. But it sounds good. Tell me about it.
I think it's mostly plastics based, but the follow up to that is related to that. What are your thoughts on how ocean microbial life is affected by commercial fishing and plastic pollution, and how does that affect the ocean population at large? Oh
wow. Okay, so lots of parts to that. Thoughts on how ocean microbial life is affected by commercial fishing and plastic pollution. Okay, so let's start. Ocean microbes and commercial fishing. There are a lot of microbes out there. And yeah, I am not the microbial person. I guess I sort of take the long view. I think they're probably, they're fine with ocean fishing. I don't think the microbes are going to change that much because of fishing. I could be wrong. But I think that is sort of going on a level where they're like the microbes, and by that I mean like bacteria and archaea. I don't think they mind the ocean fishing, but plastics is sort of interesting. We were sort of something that we observed in my lab just. So we're measuring a bunch of phosphorus, and we store, when I go to sea, I collect samples and store, oh no, guys. I store them in a plastic bottle, and we've noticed that phosphate sticks to the walls, and so that caused us to try and look at this and figure out the timescale that it happens on. And phosphate does stick to the walls of plastic, like HDPE is usually what we collect our bottles in, high-density polyethylene. I think that plastics may end up sort of collecting phosphorus, but phosphorus is one of these things that's also kind of scarce, and so I think microbes would basically immediately colonize colonize any plastic surface where there is
I don't know how
phosphorus.
You lived through that. That was amazing.
Like straight muscle memory.
There we go. Sorry, we celebrate science and gaming. There it is. Another one. Sorry, phosphorus sticks to plastic.
Yeah, that's it. And then I think the microbes see that and they go, and they chomp on. So it basically creates a substrate, like a surface for them to grow on, which is sort of interesting. And but I don't think we really understand it very well yet. So I think there are a lot of people working on this question and are really interested in it. I'm not trying to say that it's not important or it isn't, plastic isn't detrimental to the ocean, because I do think it is. But maybe for bigger organisms that would ingest it, but most phytoplankton aren't going to accidentally ingest.
I mean, so that's a good question. So microplastics, like from cosmetics and stuff, that's micron scale. I mean, that's still not going to affect plankton then?
I mean, I don't know. It's possible. But I don't. Would it get stuck in one of their transporters? I don't know.
I mean, it gets stuck in our lungs, so.
Yeah, right.
Turns out not great stuff.
Not great stuff, for sure. So yeah, I think, every meeting that I go to, there are more and more sessions on this, but I guess I haven't seen any reports yet of how this impacts plankton. That isn't to say that it doesn't. It's just going to be really hard to demonstrate robustly. You know what I mean? Mm-hmm. So, and I guess, yeah. do we know if we haven't? I was going to say we haven't seen any detrimental effects yet, but the time scale over which we've sort of been evaluating phytoplankton is probably in a robust way is probably since since we've started adding plastic to the ocean. So
still pending. All right, there's a link for the Rotten Tomatoes on Seaspiracy. It came out 5 days ago, so you'll have to forgive Angie for not having seen it yet.
I have seen the ads on Netflix, but I have not watched it.
Yeah, but if anyone's interested, it's on Netflix. I think it primarily focuses on plastics in the ocean.
Okay.
All right. Reese's Pieces wants to know, what type of mathematical models do you mostly use?
Yeah, I don't usually. So I make the hard geochemical measurements, and then I work with modeling colleagues. So some of the things are inverse models or forward models. Some I do, my favorite is a 2n number mixing model, like you use to calculate your GPA. But that's kind of what we do. But I do have colleagues that I work with, but yeah, they focus on the modeling and I focus on the measurements.
Nice, very fun.
Why can I not get out of
up arrow?
my stuff?
Are you pressing up? There you go. It's been a while.
Team effort again.
Yeah, Bronco nailed the door rage. You didn't rage quit yet.
Yes.
3 or 4 more doors. We're going to lose it.
So long as you guys are helping me out.
That's awesome. All right. Neela's1 wants to know, how do you think the future of climate change will affect processes such as the nitrogen cycle? Do you think there will be a major change, and could this affect larger biological processes?
Oh man, such a good question. And this is sort of what keeps a lot of oceanographers really busy. So there are a couple ways that climate change, and in particular warming temperatures, affect the ocean. One is it affects stratification. And so that is important because, the more temperature stratified the ocean is, the harder it is. The deeper waters are nutrient rich and the surface waters are nutrient poor. Photosynthesis goes on in about the upper 100 meters and it pulls nutrients out, and those come from often the deeper part of the ocean, so physical mixing with the subsurface. Warming temperatures make it harder to mix between the subsurface and the surface waters. And so there's a lot of concern that increased stratification will basically limit phytoplankton growth. So it'll be harder for phytoplankton to get the nitrate and the phosphate from the deep ocean. And if that happens, then that's sort of a positive feedback in the negative sense for atmospheric CO2 concentrations to increase. Yeah, that's no bueno. So there are a lot of people working on that. There are also a lot of people who work on ocean acidification and how phytoplankton respond to changes in, atmospheric CO2 concentrations, right? And so like the enzyme Rubisco, like we said, that carries out photosynthesis is adapted to actually higher atmospheric CO2 concentrations like existed when the enzyme evolved, right, billions of years ago. And so people wonder if plants will be more efficient, right, at higher atmospheric CO2 concentrations. And so some climate skeptics use this as sort of an argument to say, well, plants will be more efficient, it'll be okay. But there are a lot of other trade-offs, and we don't think that's necessarily the case. So, but it's really complicated, right? All these are really nonlinear systems, and so there are no simple answers. But there are a lot of people working on all these different aspects to try and understand how it'll work out. Well, maybe not a satisfying answer.
Yeah, well, no, we're running the experiment live, so we're going to get an answer.
That's right. Yeah. We're watching to see where phytoplankton do better and where they do worse. And,
yeah. That's fun. Mildly related question. What's the difference between phytoplankton and plankton?
Good question. Phytoplankton are the ones that are photosynthetic. And so they fix carbon. So there are single-celled plants that live in the ocean. Whereas plankton are just anything that floats in the ocean and can't control the direction it goes. So not swimmers. So they just are carried along by ocean currents. They might be able to sort of vertically regulate their position in the water column vertically through some fancy tricks. But yeah, they don't have tails and fins. They can't swim.
All right. Reese's Pieces follow-up. Do you work with any biomathematicians? On a related note, have you heard of FSU's biomathematics program?
Should I be? It sounds like somebody's like, lady. No, I have not heard of FSU's biomathematics program. Sounds interesting. No, I do not. I don't think I work with anybody who calls themselves a biomathematician. People I work with are modelers and molecular ecologists, so people who look at DNA and RNA in the samples to see which organisms are there and what's being expressed. And then also some people who look at the chemical composition of the organic matter that we find, in addition to me measuring the isotopic composition. So those are the people, but maybe I need to be talking to folks at biomathematical modeling. I don't know. Tell me about what would you do for me? Tell me what we can do. I want to know. Make my life better.
All right. Triboluminescence.
Biomath program is great. I believe it. Okay.
Triboluminescence, which is an awesome name, by the way. How is nitrogen at the bottom of the ocean different from nitrogen at the top?
Oh, great question. So nitrogen at the bottom of the ocean. So on average, the ocean is 5,000 meters deep. And the bottom 4,000 meters are just, they're real cold. And they formed at the high latitudes, like the Southern Ocean and then the North Atlantic. So one, it's cold. There's a lot of nitrate down there. And in the surface ocean, it sort of comes from different places. So if you take my chemical oceanography class, we talk about how ocean water masses are born. So they are born when they subduct from a certain location, and then they go through circulation. They go all over the ocean, and then they are sort of reborn. They die and are reborn when they reach the surface ocean again. But in general, there's no nitrate in the surface ocean because phytoplankton eat it all. And then at depth, other microbes take dying phytoplankton biomass and respire it, just like we respired our dinner. I'm gonna die. You guys. And so there's a lot of nitrate at depth, but not in the surface ocean.
All right. Ufluidic says FSU biomath program is great. So
I can't wait.
Take it from
Somebody
Ufluidic.
send me an email. Yeah, send me an email about it. Tell me all about it. I can't wait.
Yeah, somebody can do something for you. Or synergistic growth. Mm-hmm. All right. Do you have a favorite ocean organism, plant, or creature?
Oh yeah, I mean it's gonna be Trichodesmium easily. Yeah.
Okay.
Still on Trichodesmium. If you weren't here earlier,
there's a clip. Don't worry. Trichodesmium is the go-to. All right.
Yeah.
Let's go with something bigger.
Geez. So it's pretty awesome when we're out at sea. We have a lot of dolphins that swim along the bow, and that's just fun. I mean, how do you not love a dolphin, right? So dolphins are great. But scientifically, I don't have a justification for any love of megafauna other than just they're adorable.
We're scientists. We can still like things just because. It's okay.
I do. Yeah, it was awesome. Every time there's dolphins that come swim alongside of the ship, everybody you just can't help but love it. It just makes you happy. You go stand next to, you just watch them. We take videos. Yeah, it's great.
So have you seen like massive whales breaching like you've seen?
No. Most of the time we don't have megafauna. It's not uncommon to have dolphins. Dolphins happen a lot. In the eastern tropical South Pacific, we had a whale shark when we were nearer to shore, and that was unusual and pretty awesome. Tuna, seen tuna. But
no!
I knew it was so
close.
I didn't say, I went in instead. But yeah, a lot of flying fish out there, but not a lot of megafauna. So I have not seen whales breaching when I've been out at sea. That would be great.
All right, we are 40 minutes in. It is time to do a prediction. So everyone, brace your internet points. So if you're not following us, click follow right now. You'll get your 300 standard internet units, and you can gamble them on a prediction. And so we're going to pop up a prediction right now. The question is, we're going to talk volume of water. And so Mississippi starts in Minnesota, dumps water into the Gulf of Mexico through Louisiana. And so the question is, how much more water is evaporated from the Pacific Ocean versus enters from the Mississippi? So you're comparing on an annual basis, the Mississippi dumps a volume. How much more evaporation in the ocean occurs than that volume? Is it 40 times or is it 400 times? So click your guess right now, 40 times or 400 times more. So again, this is volume of water being dumped from the Mississippi River into the Gulf of Mexico versus the total evaporation that happens from the Pacific Ocean. Is it 40 times different or 400 times? No, make your bets. Place your bets now, 40 or 400. Again, click follow. You get 300 free internet points that are entirely arbitrary, but you can gamble with them, so you might as well use them. See, I can't bet internet points. I wish I could. They give me infinite internet points.
Oh, because you're the host. Yeah, they should.
Yep. Yeah. Well, that means I could just click request a factoid and take a drink over and over again. Just break the system.
Ken's living the dream.
I am. It's too much power for one individual. I should not have this position. All right. While the prediction's going, does anyone have any questions? Throw them in chat right now. Ooh, we got a 30 versus 180. 10 seconds left. Make your guess now. Spend your arbitrary internet new units.
I'm never going to clear this level, guys. It's going to be rough.
That's okay.
Over. Jeez.
Ken's the 1%. You are absolutely correct. When it comes to made-up internet units, I am super rich. All right, so the submissions are closed. Angie, what's the answer? How much more evaporation from the Pacific Ocean versus water being dumped in from the Mississippi?
It is the bigger number. It is the 400.
400 times more. And so the Mississippi collects a huge portion of water runoff from the U.S., right? And it's still tiny compared to the evaporation that occurs in the ocean.
Exactly. And so one of the points here is that, evaporation over the oceans. So the oceans are warming, right? And small changes in the rate of evaporation over the ocean will lead to huge changes in water fluxes, right? Can significantly change the amount of precipitation that happens downstream. So, so potentially very consequential. Oh God.
I mean, that's a massive amount of water. Like we can't even conceptualize how much that is. And it's such evaporation is such a subtle thing that just
it is. Although in Florida, not so subtle.
Good point. But congratulations for all of you. Oh, say that again.
What was the ratio?
So 9 people guessed 400, 2 people guessed 40. The people that guessed 40 bet 30 internet points, and the people that guessed 400 bet 180. So the payout on 400 was 1.17 to 1. So yeah, the Twitch balances the betting, so that's kind of fun. All right. Nielsus wants to know, Nielus, what's the best cruise you've been on?
Oh man, I can't pick. That's tough. That's like picking your favorite child. Not necessarily. There could definitely be bad cruises. I have gotten to be on a lot of fun ones.
Have you done the Darwin
recently,
cruise?
No, I don't know what that is.
There's a cruise that recreates the Darwin, the Journey of the Beagle.
That doesn't sound like science. Yeah. It doesn't sound like I'd go get any good samples. Yeah, so I had 2 cruises in the Eastern Tropical South Pacific that were like 5 or 5.5 weeks long, something like that. So we left out of Chile and went and sort of drove around the middle of the South Pacific, or as far as we could get. And those were both really good. We learned a lot of really interesting things. And they went pretty well. So those were good. I've done some cruising in the Gulf of Mexico lately, and those we found really interesting stuff, although I'm an oceanographer, guys, and I get seasick. Like, I am barktastic, and my best seasickness medication was off the market for a while. I was like, no, we have a problem. And,
follow up, what's the best seasickness medication?
Medicine?
Yeah.
Well, so it depends on the person. So what works for me is the patch. So it's scopolamine patch. It messes with some people's depth perception. So some boats won't allow it because people have almost walked off the back of a boat. That would be bad. That doesn't happen to me.
Side effects include, holy crap.
Yeah, exactly. Don't do it. So yeah, some boats, the captains aren't a fan of people using scopolamine, but it's my best friend. And then, like, a couple years ago, it was off the market. And I was like, what? I don't understand. It's been on the market forever. Like, what? And I tried some other stuff, and I didn't have the medications balanced right, and it was not good. So that was no fun. And the boats that go out there are barftastic. So, like, Coast Guard people go out, and they can't at all. So, yeah. Oh, my star. Oh, Lord. Oh, I lost the star. That's tough. I went down with the star. So yeah, they're all good cruises.
Oh man, that's you've inadvertently answered a subsequent question. Is oceanography a good career if you get seasick? The answer is good enough.
Yeah, good enough. That's right. I don't go out that often.
As long as you don't care about depth perception, it's fine.
That's right. That's right. It's a good way to lose a few pounds. You know? No, it's not. It doesn't. The pounds don't stay off. You come back and it doesn't actually help anything. Yeah, it doesn't really matter because most oceanographers actually get seasick, and pretty much everybody's on medication. Even if they act like they're not, they are. They're lying. It's a front.
So do people get used to it? Do they?
Some people do. Yeah, the longer you're at sea, the more you get used to it. And then there are some people who just don't get seasick, who are just fabulous like that. But most people do, and most people take medication, and most people figure it out. So it's not so bad.
That was amazing. All right. Triboluminescence. Here's an easy one. How did life originate?
Oh, yeah. Okay. Well, if you figure that out, I think you get a Nobel Prize.
At least.
So I think there are a couple, theories. Biofilms and microbial mats are popular ones. So microbial mats, like, think of the scuzz around your bathroom sink or something when it hasn't been cleaned in a while, right? And the pretty colors that you see, like, there are a couple places where there's still what we call microbial these stromatolite mats. And so the Bahamas is actually one place, and then Western Australia has some stromatolites. But we think we see them in the fossil record going back a couple billion years, although that's kind of hard to, robustly demonstrate. But some people also think hydrothermal vents, because there's a lot of reducing power that evolves there, and there's sort of the last common ancestors. So the molecular ecologists have sort of done a tree back in time, and some of the microbes that sort of retain the oldest properties are most similar to the microbes that we find at hydrothermal vents and at places like Yellowstone hot springs and things like that. So those are some of the working hypotheses, but really tough to figure out. But nitrogen fixation had to have been one of the first metabolisms. So because they needed nitrogen and there wasn't enough in the environment a couple billion years ago. And so we do think nitrogen fixation was one of the first metabolisms.
So if you had to bet imaginary internet points, you would say hydrothermal vent?
That's a tough one. I mean, sure. Yeah, I go with hydrothermal vent. I kind of love microbial mats. I think they're awesome. But I think probably right now, just because I think they're fun, but I think probably the strongest evidence is for hydrothermal vent. But it's all pretty arm wavy at this point. So.
Yeah. Makes sense. All right. Bronco wants to know, are there any plans to having higher cost point redemption? I don't know, because we only do... I mean, we're going to do streams like every other week, especially when we can do it in person. You can't build up that many points, but if you guys have any ideas for point, spending internet points on things, we are happy to consider them. I won't promise we'll commit, but we'll definitely look at them. Shiloh wants to know, what's your zodiac sign?
That's hilarious. I am a Sagittarius.
How does that affect oceanography?
I don't know. You tell me.
The answer is not at all. There's an awesome...
I am not aware of how it affects oceanography.
There was a physics professor. I mean, so the assumption behind the... Yes!
I get a beverage.
Yes. No, cheers to that. We don't here, I'll do it. We're going to do the formal take a drink. Redeemed by Ask a Scientist Gaming. Cheers, Angie. Congratulations beating the first castle. The first of the Bowser kids. Well done. No, but there was a physics professor that basically, I mean, presumably the star position affects your existence somehow, and the only way it can do that is through gravity. But you can do the math, and like, my gravitational pull on you right now 5 miles away is higher than a star that's, light years away. So it's absolutely nonsensical based on what we know of modern physics. So, yeah. All right. So zodiac sign. That's probably the easiest one of the night, huh? Let's just keep in mind, we went from origin life to zodiac sign. Welcome to Ask a Scientist Gaming, where anything goes. Oh, Reese's Pieces has a fun one. What's the most outrageous thing that has happened out in the field? Like you have a riskier job than most of us researchers.
Yeah, let's see. So yeah, the oceanography, makes it. I just lost my shirt. So actually, on one of the Gulf of Mexico cruises in 2015, we rescued somebody. So our cruise got in early, and but not early enough to have crew there waiting for us at the dock to help us unload and tie up the boat. So we sort of sat out in Tampa Harbor. So I've gone in and out of St. Pete with my colleagues at the University of South Florida, and so we were in our boat. And it was a short cruise. We were only out for 3 or 4 days. But I was out with 2 other faculty, and we had a bunch of students, including some FSU undergrads and some graduate students. We were parked in Tampa Bay waiting to go in early the next morning. And the other 2 faculty members and I were like, we're old, we're tired, we're going to bed. And then there was sort of an alarm call from the bridge, but it wasn't, Yeah, so it was a man overboard call from somebody else, another boat in the area. And so we went to go help look for the person. And the undergrads, they're never tired. So they went and stood on the side of the ship and were looking, and the ship had its spotlight out and stuff like that. And they found him. So he had been drinking in a small boat and had fallen off the side, and he was probably not going to last much longer. So that was pretty amazing. We also, but people, there are pirate lockdowns, so pirates are a real thing out there, and so boats don't go to the Indian Ocean. We can't go there because the threat of pirates is too real, too dangerous. So, I mean, we can tell these stories all night.
Well, follow up on that. Actually, before I do that, so Ninja Physics tried to post a link earlier. Apparently, there's legislation at the U.S. federal level. It's a bill to address plastics pollution. And so there's a link in chat if you guys want to take a look. Yeah, that's something we definitely need to deal with. We are kicking that ball down the field until it becomes a major problem, but it's coming. Like, there's no question.
Yeah.
Also,
yeah, stop buying plastic. Stop buying stuff.
Reusable, renewable, recyclable, classic mantra. But honestly, if you're watching Ask a Scientist Gaming, we're probably preaching to the choir a bit here, but tell your friends and family. More importantly, family, grandparents. All right. Shiloh's calling us out on the Zodiac. He says Sagittariuses are more statistically inclined to become oceanographers. So that makes sense.
Way. Show me that. I want that reference.
Yeah, feel free to whisper me a link to that one. But I don't know enough about it to argue, so we'll say it's true.
How would you even know? How would you get everybody's birthdays? I'm fascinated. I want to know. I want to know the methods.
That's amazing.
I don't have enough muscle memory for this level. This is tough. Okay.
All right, so follow up to the last question. This is one from the list. Is what's the biggest mistake or accident you've seen?
Right. Well, let's see. Thankfully, there haven't been any real serious ones on my cruises. Not because we're special. I think we've just been lucky. But people lose a lot of expensive equipment over the side. That's a problem, especially if NSF, the National Science Foundation, owns it. Don't do that. I mean, people get hurt out there. So there's a story from a cruise in the 1980s or 90s, and there was a boat in the Pacific, I think. So U.S. research vessels used to do swim calls. So they used to let oceanographers jump in the ocean and go swimming out in the middle of nowhere. And they don't anymore because the last time they did that, they were having a barbecue on the back deck. They threw the leftovers in the water, so sort of chumming the water. And then they went swimming, and a shark came and literally ate a limb off one of the science parties. And so no more swim calls for U.S. boats. So we don't NSF-funded boats. So that, I mean, and then there's some other good ones, right? So better than that, because that's terrible. We don't want that to happen. So this isn't good either, though. So, but nobody lost a limb. So I think, well, I know the institution, I won't say, but they had a boat. And so some schools have boats. FSU has a small boat that mostly hangs out in the coastal Gulf of Mexico. But this was another boat at another, let's just say, Florida institution. And either the captain or the first mate passed gas. And it was so bad that both the captain and the first mate left the bridge, the steering space, right? And they ran the boat aground on coral. So that's bad. So, there are all kinds of ways things can go wrong.
How expensive was that fart?
I don't know, but it was a big problem. So it hurt the boat, but then also coral reefs are protected, like the government doesn't like that.
That's no joke. Yeah,
that's no joke. Like, you are not popular anymore. So I think it was pretty consequential for the institution and the boat and the captain and everybody involved. So not good. So there are all kinds of. So you really have to be intrepid to be an oceanographer and ready to problem solve. So we've also gone on cruises, and then you can't get your gear. We've had colleagues who. We were trying to go out of a city in Chile, and some other colleagues had been in Antarctica, and they were trying to get their gear through Santiago. And apparently Chile has it's basically different countries in terms of trying to import and export gear. It's really hard. And they couldn't get their stuff out. So some of our colleagues just couldn't go. So you have to know customs and how to get stuff through customs. So you just have to be willing to sort of roll with the punches and be resourceful and sort of expect that not everything is going to go exactly right, and figure out how to adapt on the fly and build stuff out of whatever is around on the boat if you can't get all your gear to get your science done. So it's kind of MacGyver. It's very MacGyver for those. Since we're going with 1980s references.
To be fair, I would put most research under the umbrella of MacGyver.
Yeah, that's right.
You have to be a plumber, an electrician.
And you can't go to Granger on campus and get supplies. We have MacGyvered a lot of things.
Oh, follow up from Shiloh. I didn't read the next comment, but Sagittarius thing was entirely made up. We were fairly confident it was.
You just broke my heart. Okay.
I mean, what's crazy is you might have some kind of like birth rate difference between coastal cities, and that like it could happen. You could imagine a scenario where that would occur.
Come up with a legitimate explanation here.
Yeah. All right. Bronco wants to know approximately how much of the ocean is considered unexplored / unknown.
Oh man, such a great question. I mean, I think most of it. The ocean floor the ocean takes up 70% of Earth's surface, and we explore almost none of it. Most oceanography happens in like the upper 100 meters or something like that, and not even the deep ocean. And on average, the ocean is 5,000 meters deep. So we know very little. And then there are a bunch of people who are working on marine sediments, which are really interesting. And actually, there's a ton of microbial life down there. And so, yeah, lobby your Congress people to give more money to ocean science. It's amazing. We don't understand most of it. So, and it's right here, right? Like, we don't even have to go to another planet. Like, people look for analogs for life on extraterrestrial life in these extreme environments, right? So down in hydrothermal vents or in microbial mats or all kinds of things. So, yeah, oceanographers will tell you we know nothing and we need more money to study it. So that's going to be our answer.
That's a good mantra. All right. Ninja Physics wants to know, are there differences in phytoplankton that are based on varying latitudes?
Such a good question. We think generally yes. So we think things like, there are definitely phytoplankton that have temperature preferences, right? And so things that live in warm waters, things that live in cold waters. Life tip for those of you considering oceanography, you should want to study things that live in warm waters and go to the tropics. I'm just saying.
That's amazing advice. that is perfect.
I mean, I'm not saying I don't want to go to Antarctica and check that out. That sounds amazing too. And the Southern Ocean is fascinating and climatologically super important, more important than the low latitude. But also calmer seas. If you're barf-tastic, pick a low latitude study site. So yes, but they're like, my favorite Trichodesmium definitely prefers warm waters, like 23, 24 degrees C and up. A lot of diatoms they're found in these cold water environments. So Southern Ocean or coastal upland and things like that. So yeah, for sure. There's a lot of so it's not so much latitudinal preference, but it's other environmental variables that correlate with latitude, like temperature. You guys, I'm never going to pass this level. Sorry.
No worries. We'll do a bet on it at some point. When you get close, the next one we'll do a bet on it. All right. No, you need the pressure. It's going to work.
Mm-hmm.
All right. Focus, Chelsea wants to know your favorite time period in Earth history.
Oh, Chelsea, you do that to me. Yeah, she's going to see if I remember anything.
The answer is right now because of Ask a Scientist Gaming. So
yes,
easy.
Ordovician. I'm going to go with Ordovician. Yeah.
Can you expand on what that means? Also, you can tail whip that guy or cape whip.
Yeah. If I can button mash my way through that. So they're in the Paleozoic. A lot of really interesting life forms were evolving in the Ordovician. So Silurian and Ordovician, really great time periods. A lot of life that looks more like what we are familiar with was starting to evolve. And so land plants and not the dinosaurs, but proto-dinosaurs. Oh, I've got to fly up there.
No. You can run up that. So if you run at that little corner as fast as you can, you'll walk up the side.
Oh, this is where I need my brothers. Oh, God.
Yeah,
Okay.
just keep running.
My brothers at this level. Yeah. All right.
Now you can jump. Oh, there you go.
Oh, and then you fly through the coins. Oh, yeah. All right. Okay. I haven't made it to this level with my kids. I haven't been back to this level in I don't know how long. So, okay.
No worries.
Yes. Chelsea, what's your favorite?
Based on her answer, it's the Silurian. She was offended by your earlier answer.
I'm sorry, Chelsea. You're right. It is true. The Silurian is better. I defer to Chelsea on all things. She's correct.
All right. So Triboluminescence has followed up on their question of what's the origin of life with how realistic is the movie Life Aquatica? Shiloh says marginally more realistic than Sharknado. What about Deep Blue Sea? These are all quality documentaries filmed in real time. How realistic was Life Aquatica? Have you seen it?
No, I've seen that, you guys.
Oh man. The movie question's not hitting. Have you seen Sharknado?
It's not. No, I've heard of it, but I haven't. No.
Yeah, I think there's 6 of them now.
Yeah.
so worth the viewing.
Yeah, I'm sure. No, I am a parent of 2 with a partner who's also an actor. We watch very little TV. And there's a lot of My Little Ponies that happens in my house, but I don't necessarily watch it.
Well, so I'll follow up with one of my favorites. Of the movies that you have seen, are there any that get your discipline right or wrong?
I'm not aware of so no, because I haven't seen any of these movies. So apparently, I'm like, I haven't seen any that cover my discipline. And apparently they're out there. I just haven't watched them. So.
Give it time.
Yeah,
we'll find them.
The kids are getting older. It's good.
Exactly. They'll be old enough and someday we'll get to go back to movie theaters, which will be amazing. What about Jaws?
I cannot speak to charismatic megaphone, so I don't know. That's not my expertise.
So the author of Jaws ended up regretting the book later and became a shark conservationist because it made the public so actively opposed to sharks because of how scary the movie was.
Right. Yes. So yeah, poor sharks. Yeah.
Bracco, how good is The Abyss? We have established Angie does not watch movies. Let's move on from movie questions. Let's move on to marine biology related music. I
don't know. Indeed. All right, let's try.
Chelsea recommends Meg. Gotta watch The Meg. It's about a megalodon hiding at the bottom of the ocean.
I saw that on a plane once, and that was not right. Yeah, so I think I saw that. I didn't listen to it, but it was like on somebody else's screen. You know what I mean?
We got to an answer.
Yeah,
that's awesome.
And the answer is no.
That's amazing. How good is The Abyss? Citrine C wants to know, do you prefer water off the east or west coast of the U.S.?
Oh, I mean, that's not different. So they're just really different. So there is upwelling off the west coast. So they're just very different environments. So again, this is like a pick your favorite kid. Can't do that. The east coast of the U.S. is shallow continental shelf, and so has very different ecosystems. So just really different processes going on in both systems, we think. It's for different dynamics. So the west coast of the U.S. so Eastern Pacific is upwelling, very seasonal, and lots of diatoms. Very productive. East Coast, continental shelf, lower nutrient, trichodesmium. So tricho on the East Coast. Not a lot of tricho on the West Coast. So
East Coast wins.
I guess so, yeah.
There you have your answer. Awesome.
Yeah.
Ninja Physics, thank you for continuing the gift stub from Hamsterlicious. Again, we appreciate subs. Again, pays for beer. If you are going to sub, use your Amazon Prime, attach it to your Twitch account. Then you can have Jeff Bezos pay us rather than you guys. Use your free Amazon Prime associated with Twitch account to do subscriptions. So, yeah, thanks, Ninja Physics. Pleasure to have you around asking questions, and thanks for the sub. All right, Bronco wants to know how likely, follow up to the Meg, how likely is it that a sea monster exists and we just haven't found them yet?
Oh, I think you should hold that out in your heart as something that's out there.
So the answer is no, but keep hope.
Keep hope alive. Yeah, I mean, why not, right? We haven't seen a lot of it, so yeah, I don't know. Go ahead.
So
I mean, we still find stuff we've never seen before all the time.
So is there
coelacanths are still down there? That's for Chelsea. Coelacanths, living fossils. So yeah.
I mean, is there a relationship between like depth and the size of the animal? Like, there's got to be something that prohibits deep sea big creatures existing.
Right.
I'm guessing.
Yeah. So, yeah, I mean, fundamentally, they just need to eat, right? Mm-hmm. And so this gets back to, when I say the east and west coasts are different environments, and there's upwelling off the west coast, that means there's a bunch of nutrient-rich deep water coming to the surface that fuels phytoplankton, that fuels a dynamic, rich food web, right? So you've got zooplankton and fish and bigger fish and sharks and stuff like that. And so my guess would be that you would find those things maybe near coastal margins or these upwelling systems where you've got a lot of nutrients around. But usually those things that are big and at depth, they have such slow metabolisms and they live so long that I think they could probably do anything. But again, if there are any charismatic megafauna people that are watching this, like, I'm not a biologist. I'm definitely not a megafauna person. And so I don't know anything about big things. So I could be wrong. But I think, yeah, I think they could be anywhere, but maybe more likely to be in a coastal system or something like that, where there's a bunch of food falling down to them to eat.
All right. Sorry, going back, catching up on a few. Triboluminescence wants to know, why haven't plants evolved to fix their own nitrogen?
Well, such a big question. So I think this gets back to this.
Scuzzbot, thank you for the gifted sub to Triboluminescence.
Oh, man. So two parts to that, right? So one, the nitrogen fixation genes are just this monstrous set of genes, and so they're really cumbersome. So this gets back to this black queen hypothesis that somebody's got to take it for the team. And it probably, when it evolved, probably the first plants, right, they were cyanobacteria in the ocean, and maybe they were cyanobacterial diazotrophs, so that they could, cyanobacteria who could fix their own nitrogen. And so maybe that's how it started, right? So maybe they were at the beginning on land, so it's symbiotic nitrogen fixers, right? So there are microbes that live symbiotically with nitrogen, with plants, but I went to a conference a couple years ago. It was on nitrogen fixation, and you guys, like, seriously, Monsanto is trying to engineer nitrogen fixation into plants. So it's coming soon to a plant near you. So they don't have to dump a bunch of fertilizer, right? And so then they get out of environmental trouble. Yeah, I know. But then, like, there was a guy at Harvard who's trying to do it too. There are groups around the world who are working on this. So at Harvard, and I think there's somebody in Spain or somebody like that. But yeah, so there are people trying to bioengineer this, and it seems like they're not insignificantly far along.
Yeah, I think I know who you're talking about at Harvard. I think Dan Nocera is working in that area. Oh, there's the tape. That is the end of the level right there.
I was so close, right?
Yep.
Didn't happen.
So close.
Just want to stay here, I guess. My identity is not tied up in my ability to work somewhere. You can all judge me for it. It's okay.
Reese's Pieces, is life really better under the sea?
I don't know.
Shout out to The Little Mermaid, which I've seen many times fairly recently.
And also homeless children.
Yeah, exactly.
Yeah, so I don't take submersibles down, but I have some colleagues in the department who do, which is pretty awesome. But I mean, the ocean's pretty awesome. It's pretty awesome being out at sea and being out in the middle of nowhere where, all you can see is ocean. It's pretty cool. So it is like at sea. I'll say life at sea is better.
But under the sea?
I don't know. I don't go under the sea. So it sounds great, though. I don't know. It sounds pretty amazing.
Based on the song, I'm led to believe it's awesome.
That's right. I think, according to Ariel, yes. Actually, she wants to be on land, so, you know.
That's true. Sebastian was the one. Sebastian,
yeah.
Yes.
That's right.
Got to get our Little Mermaid lore. All right. Niece Boulder wants to know, when and why did bananas become your catchphrase? Did you know you have a catchphrase?
It sounds like somebody, Nice Boulder, yeah, somebody who was probably in my classes. I don't know when that became, but yeah, so I definitely say, this is bananas, right? So there are definitely some of my former students from my classes in here. And so scientifically speaking, there are things that are bananas. Absolutely true.
One thing I learned fairly recently is phrases like that, bananas, explaining that to a 3-year-old, it's nonsense. When I describe something as bananas, it breaks their mind. So that's awesome.
Oh, they just crack up. They love it, right? Mm-hmm. Yeah, when you're trying to get a class also to, like, they're bored and they're dying. And try and, like, you know, get their attention. So yeah, things are bananas. Like trichodesmium, usually they hear it in the context of me talking about bananas. So it's crazy. Shouldn't
awesome.
do these things. It does. It shouldn't fix both nitrogen and carbon because oxygen is generated from photosynthesis, but oxygen permanently, irreparably denatures the enzyme that is required to carry out nitrogen fixation. So how did they evolve to happen in the same organism, in single-celled thing? Like, come on, get out of town. It's nuts. So people still trying to figure it out. We don't totally understand it.
Oof, that was close.
No, it is gonna be close. Come on, Yoshi.
Idiom. Thank you for the link, Scuzzbot. That was in fact an idiom.
You guys, I'm gonna make it. I cleared.
Oh, we did it. Well done. Cheers to that. Mm-hmm. It's the little victories. Citrine Scene wants to know, when was the modern nitrogen cycle established?
Oh, that's a good question.
Shiloh, thank you for the follow. Enjoy your 300 standard internet units that we will bet some point soon. Sorry, when was modern nitrogen cycle established?
So by that, I'm going to assume, like, when did we understand the nitrogen cycle? I'm not sure we understand it yet, but I think it's been operating. Oh, Yoshi, come back. I think we think it's been operating sort of in the current form probably for, that's a great question. I mean, the processes have all been around for hundreds of millions, if not billions of years. So I don't think processes are sort of new to the scene. Microbes have oh God, I'm gonna die. I think the microbes have been around for a long time. Our understanding of it, we still don't get it. So we're still trying to understand it. So, but the microbes got to figure it out.
All right, so Ninja Physics has requested a factoid, and I'm going to tie this one with Citrine C's follow-up question. What is the most scientifically bananas thing you can think of off the top of your head?
Oh, come on. Okay, well, go ahead.
Something that blows your mind or blow our mind.
Okay, so just to talk about something other than trichodesmium, because we'll change topics. I don't sound so much like a broken record. There's a lot of nitrogen, right, in the Gulf of Mexico and in the Mississippi River, right? We know that there's all this farming. Mississippi River drains all these farmlands, right? And Monsanto is trying to get nitrogen fixation in plants so they don't have to apply fertilizer. So in the meantime, there's all this fertilizer in the Mississippi River that comes out. So we tried to look for it in the Gulf of Mexico. We tried to look for all this nitrate from the Gulf of Mexico, from the Mississippi River in the Gulf of Mexico, and we couldn't find it. you don't see it coming out. So all the nitrogen that leads to the dead zone and stuff like that. And this actually is consistent with a lot of other people's work in terms of where river runoff goes. But we were still surprised because there's so much nitrate that comes out of the Mississippi River. It's not ending up in the deep Gulf of Mexico and leaving the Gulf and going to the Atlantic Ocean via the loop. So I think that's pretty wild. And so I had an undergrad who did her honors thesis with me a couple of years ago, Sammy Howe, and she's great. I think she works for the DEP. But yeah, we looked for it in a bunch of deep water Gulf of Mexico profiles. Couldn't see it. So the nitrate coming out of the Mississippi River is isotopically distinct. So it's geochemically different from what is in, from what's in, I was so close, right?
Yeah.
Sigh. Oh, game over. That's rough. Well, luckily, we're here all night.
We did not figure out Game Genie codes ahead of time. We should have. So it goes. All right, so nitrates are, I mean, there's runoff from, farms all throughout the entire basin, and none of that makes
Minnesota.
it. Yeah, and none of the, I'm from Minnesota.
Yeah, that's why I gave you a shout out.
Do you hear it?
No.
All right. Oh, we're back to this level. Your arch nemesis.
I know. Back to yeah. So, or this awful level.
So
if I got any better.
So none of the nitrates from the Mississippi make it in the ocean, which is kind of crazy.
Yeah. So there's been a bunch of recent modeling work that suggests that a lot of that is due to a lot of it stays on the shelf and stays really basically near the shore. And so it has to do with the width of the shelf and also the latitude, because the Coriolis force varies with latitude. And so there's all those coins up there. See, I am not living up to my potential on this level. I think you'll have to forgive me.
It's okay.
At all. This level is not my friend. So that, and then also the loop current, which is what comes through the Florida Straits and the Yucatan Channel, the water that comes through there is moving so fast that basically it doesn't stick around very long in the Gulf. It sort of comes in and does that hairpin turn and then leaves again. And so it doesn't really give that water that comes in from the Atlantic and goes back out to the Atlantic much time to accumulate any of the nitrogen that's coming into the Gulf from the Mississippi River or from other sources, or at least we haven't seen it yet. This is sad.
So I'm going to give you a hint on this level. If those things have a glowy cape, they release a feather.
Oh, okay. So kill them and get a feather.
So kill the first one and you'll get a cape right away.
Okay. Thanks, Ken. He's like, I can't watch this level anymore.
There you go. Team effort here.
Don't jump into the next one. That's the real problem. Oh, there we go.
Nice.
Okay, just spin.
All right, so I'm going to combine two of these questions. Tribo, I'll get back to yours, but so Calm Down Bronco and Reese's Pieces have a related question. So what will the next Nobel Prize in your field be for? Another one is, what is the white whale discovery in your area, and how would it affect research if it were discovered tomorrow? This might be two independent answers, I'm not sure.
Yeah. So let's go back to the first one, the Nobel Prize. I don't know if any oceanographers gotten the Nobel Prize.
So the Nobel categories are physics, chemistry, medicine, and physiology. So I guess unless it's related to medicine, it might be tough.
So I think probably the closest, so people are looking for biopharmaceuticals all the time, right? And microbes in the ocean and other ocean organisms that may provide pharmaceuticals or something like that. I think really trying to understand the mechanism that's by the ocean that is linked to climate change during the glacial and interglacial cycles, that's a big deal. And so maybe not quite Nobel Prize, but some prize worthy. Oh man.
No, get the feather. No, go back. Yes. Best of both. I'm invested in both science and gaming.
Yeah. All right. Oh, come on. Oh, stinkers. There we go. Okay.
Perfect.
Okay. Oh, I'm trying to spin.
All right, so it might be something medicinally discovered within the ocean. All right, so follow up, what is the white whale discovery? Uh-oh. We might have just lost our connection to Angela. Oh, she's back. Sorry, cut out there for a bit. All right, follow up. What is the white whale discovery in your area, and how would it affect research if it were discovered tomorrow?
So, like, the biggest deal? Like,
yes.
What do we mean by white whale? Yeah, so, like, if the big prize, right?
Yep.
I mean, that's such a good question. I don't know. I think we're still just trying to map a lot of processes. We're just still trying to understand variability and like why things show up where they do, and sort of think we understand things to first order, but then you get people who are like, nuh-uh, you know? So yeah, I think we're still just at a pretty basic level trying to map. I don't have a good answer.
Well, the correct answer is megalodon sharks in the deep sea.
Yes. Yeah, right. But again, I don't do the megafauna, so for better or worse, I can't speak to the megafauna. Yes. I think finding, the Loch Ness Monster in the deep ocean clearly is the next step.
That'd be worth some award. All right, Triboluminescence wants to know, how do you analyze water samples? Well, let's start with this. So you go out on a boat and you just skim the surface for a while. How do you collect samples, and then what do you do with them?
Yeah, good question. So I get water all through the water column. So we do have, there's usually a pipe at the front end of research boats that is sucking in water from the bow. And then that is piped all through the boat. And so we call that the underway water. So while we are steaming or underway, while the boat is moving, it is always pumping water. So we can pretty much always tap surface water inside the boat and collect it and do what we want with it. And so some people bring instrumentation and they'll be looking at O2 argon ratios, look at rates of photosynthesis, or doing all kinds of measurements on it. We usually have probes in there to monitor temperature and salinity, and then fluorescence to try and get a sense of chlorophyll. But then for the deep water samples, what we do is we have something called a Niskin rosette that is basically a bunch of plastic tubes that have lids that are spring-loaded. Oh, I'm going to die. And we have a whole bunch of them. There's usually 12 or 24 on an aluminum frame. And in the middle of the aluminum frame is sensors and a cable that is connected to a computer on the boat. And so what we do is we send all these rings of plastic tubes down to whatever depth we want to collect water from using a crane. And then we send it down as deep as we want with the tubes open. And then when we're happy with whatever depth we're supposed to, I just need to go. You need to just holler at me. Just say, go, you're almost done. Just go. Stop trying to get going. All right. So when we get to the bottom depth, then we click a button and the lids on the tube close for one of the tubes, and then we pull the collection of tubes up to the next step we want to collect samples at, and we just trip bottles that way, and then we bring it up on deck and collect water from and then filter them and do things with the water on board, or I bring it back to the lab. I freeze it on the boat and bring it back to the lab.
What do you do with it in lab?
So we measure nutrient concentrations.
Smush.
So sorry. Yeah. We measure nutrient concentrations and then their isotopic compositions.
So you do that with mass spec?
The isotopic ratios, yeah, that's with the isotope ratio mass spec. And for nitrate and dissolved organic nitrogen concentrations, we have a chemiluminescent Knox box. And that usually is strapped to the side of a power plant. So what it does is we can inject nitrate first into a heated acidic vanadium-3 solution, and that quantitatively and instantaneously reduces nitrate to NO gas, and we take that in a carrier stream of helium or N2 to a chemiluminescent reaction chamber. And what happens there is the NO reacts with ozone to make an excited NO2 molecule, and as it falls down to a stable configuration, then it releases light in proportion to the amount of nitrogen sample. And that is super sensitive, and what's great is it doesn't matter what the matrix is, so it doesn't matter if it's seawater, freshwater, or anything else. It doesn't care about the salinity of our sample, and a lot of people measure nutrients using optical spectroscopy, right? So they do a colorimetric method, but salinity changes can interfere with the absorbance. Ken knows better than most, maybe. And so this gets away from that. It doesn't matter, and you just it's very sensitive. So I love the Knox box a lot. And
that seems like such a convoluted measurement, but like photons off a sample is hard to argue with. Like that's that's such a sensitive way to do it. That's amazing.
It's really lovely, and there's no toxic waste produced. Like, so the other way to measure nitrate and nitrite is you have to have a cadmium column, which is toxic and super sensitive to oxygen contamination and a pain in the butt, and it's just terrible. And then you measure it on a UV vis spectrophotometer. Can't get that. Oh, the feathers. Oh, I'm gonna die. Can you get the feather? There we go. So yeah, it's just way more sensitive. Doesn't generate a bunch of horrible waste, and is great. And then we do other nutrients like phosphorus using colorimetric methods, but those are less pesky and toxic, and so not so bad. And then we do organic nitrogen and phosphorus concentrations in my lab, generally using similar methods. And then the isotopes, so my PhD advisor developed this method called the Denitrifier Method, which is kind of crazy. So we actually use microbes to help us measure the stable isotopic composition of nitrate, and he got a MacArthur Genius Award for it. So not a Nobel Prize, but
wow,
Genius Award. So we use soil microbes, denitrifying microbes that lack the last enzyme in the pathway, so they reduce nitrate to N2O instead of N2 gas, and that's analytically useful because N2O is a trace gas in the atmosphere, whereas N2 is 80%, and so if you have a small leak in your vacuum line, it will be a problem, but N2O, not a lot in the atmosphere. We use these microbes to reduce nitrate to N2O, and then we measure the isotopic composition of the N2O, so we get both the nitrogen and oxygen isotopic composition of nitrate. So we have to grow microbes, which turns out to be a royal pain in the butt, especially because our autoclaves are not well maintained. So this is like the bane of my existence at FSU. Yeah.
Growing the right microbes. It's easy to grow the wrong microbes. That's awesome.
Yeah, exactly.
I mean, sorry, that's horrible, but
it's seriously, it gets me down. But we've been able to make it work often enough. So that's what we do in the morning.
All right, Angie, so you are an hour and a half in. Do you want to change to Super Mario Kart? It's up to you.
Yeah, I think
Not
this level
you.
is kind of going to... Let's change it up so that nobody else has to watch.
Yeah, so if you just drag and drop Super Mario or Mario Kart, it should change up right away.
Sweet. Let's do it.
Flawless. All right, so we're an hour and a half in, everyone. Anyone just joining us, joining me today is Dr. Angie Knapp, oceanographer, studies nitrogen cycles, plays Super Mario Kart, and is happy to answer any of your questions. We should do a prediction on Mario Kart, actually.
Oh,
Will you win the first race? So when was the last time you played this?
I haven't played this version, so I played the 64 version a lot more than this one. So
all right.
Let that inform your predictions, friends. Choose wisely.
Well, we'll let you do a couple games, then we'll do a prediction on wins. For now,
it's okay. I'm not saying I'm going to get better, guys.
let's spend some internet points anyway. Standard internet units. We're going to pop up a prediction right now. We're going to gauge the amount of rain that lands on North America versus the amount of rain that lands on the Atlantic Ocean. And so, sorry, we're popping that up right now. There we go. There's the prediction. So the question is, how much more rain falls on the ocean than on North America? And so you have to take into account.
Maybe the Pacific. I think this is for the Pacific Ocean specifically.
Is it Pacific Ocean? I screwed up and I typed Atlantic.
That's okay.
All right.
No. Then you're right. Then it's Atlantic.
Okay. So amount of rain on North America versus Atlantic Ocean. Put in your guess. Is it 5 times more on the ocean or is it 50 times more on the ocean? Use your internet points now. And again, if you're just viewing, click that follow button. You get 300 standard internet units. You can gamble them on this question. Again, it's a question of total amount of rain that falls on North America versus the Atlantic Ocean. Is it 5 times more on the ocean or is it 50 times more on the ocean? Get your guesses in now. These are great questions because they're really hard to Google. I mean, we have Ask a Scientist honor code here, but also, internet points are important.
My 7th grader's science teacher tells her to Google the answers instead of, she doesn't have reading. They just tell them to go Google stuff. I was like, really? Is that where we're at? I guess. She's like, it's more useful for real life. I was like, well, I guess.
I mean, that's the dirty secret. That's what we do most of the time. The number of times I've looked up a term in Wikipedia, I mean...
Oh, yeah, daily. Yes.
Yeah, exactly.
Did I just win? Is that possible?
You did just win. Oh, man, we should have done that prediction. The Dark Horse Believers would have won.
I didn't think that was going to happen.
Congratulations. Shiloh says Toad is best, but not according to the race we just watched. I think the answer is Yoshi is best. All right, prediction. How much time is left on that? All right, prediction is closed. So what's the answer? 5 times more or 50 times more?
I believe our answer is...
I have it as 5 times more on the ocean.
Yeah. Right. So not as big as the difference that we talked about, the evaporation difference before, right? So the Atlantic Ocean is relatively small compared to the Pacific. So it is more rain over the Atlantic than over North America, but the Atlantic is a smaller basin.
So that's interesting. So it's not just geometric area. It has to do with the cycles and where rain falls. So that's presumably mountain-based and temperature gradients and whatnot.
Yeah, there's a strong band of precipitation around the equator, and then there's geography as well.
So congratulations to Ninja Physics and 4 others. 5 times more on the ocean. And again, that's a massive amount of water. That is, again, my brain does not wrap around these types of numbers.
Yeah, it's pretty hard to think about. But yeah, so again, the idea is sort of like, if the ocean is warming because of climate change, small changes in the rates of precipitation and evaporation from over the ocean end up potentially having a huge impact on what happens on land, right? Because all that moist air that's rising over the oceans or precipitating over the oceans, right? Small changes in that affect where we experience precipitation, right? And so that can mean that crops grow places they didn't used to, or crops no longer grow places they used to before. So there's actually been some interesting work in people trying to relate shifts in climate to changes in civilizations or drama, right, in the Fertile Crescent or something like that. Changes in rulers and things like that. So attributable to changes in precipitation and. Kind of makes sense, right? If your people are starving because crops aren't growing, going to be uprisings. Yeah.
Yeah, one of our fairly recent guests, Matt Howarst, he's a sociologist that studies the impact of climate change on human migration. And so they're starting to model those behaviors. So that's, that is an interesting domain. All right, Genus Boulder wants to know, what inspired you to study nitrogen?
Great question. And I think that name is Gneiss. So Gneiss is a kind of rock. So it's metamorphic rock. So it's one of my geology friends out there, I think. So why nitrogen? Good question. So I was sort of interested in trying to understand what controls climate change. And like I said, I sort of appreciated at some point that the ocean plays an important role in that. And then this, rates of photosynthesis. And so then if you care about what we call that the biological pump, so phytoplankton pulling carbon out of the atmosphere into the surface ocean and then turning it into organic matter that if you care about pulling CO2 out of the atmosphere, hopefully that organic matter sinks to the deep ocean and is then trapped away from the atmosphere for 1,000-year timescales. If you care about figuring out what makes phytoplankton grow, what makes them tick in the ocean, you probably care about nitrogen, phosphorus, or iron. That's probably what you study. And I sort of felt like nitrogen was Goldilocks. So, I also, sad, I'm on the side. So you can study carbon too. They care about that, but it's usually not limiting. But I felt like carbon was maybe, the carbon cycle was too big. There were too many people involved in that. It would be harder to sort of find a niche. Phosphorus was too small, and nitrogen seemed kind of just right. And then sort of really interesting and dynamic. And again, because it's microbially mediated, it has the potential to be just more dynamic than the phosphorus and iron cycles. So, nitrogen seemed just right. Oh.
Sorry, I had to go grab another drink.
Yeah.
Bronco, thank you for stopping by. We'll see you next time. 2 weeks. April 15th will be our next one. All right, Citrine C wants to know, coffee or tea, what kind?
Both. Depends how fired up I am on the day. So I like French roast, dark coffee, for sure. And so I grew up in the Seattle area. There's some great coffee purveyors from Seattle. I get some from Whidbey Island, from Useless Bay coffee roasters. I like their stuff a lot. And tea, I have a range of teas these days. I've acquired quite the tea collection. My husband got me some from Capital Teas, which is I think they must have a shop at one of the DC airports, so he was there, and they have something called Chesapeake Sunrise that I really like. And I didn't used to like Earl Grey, but now I'm a fan of Earl Grey. So I feel like I'm getting old because I like Earl Grey, grapefruit, and parsley, and I didn't used to like any of these things. So I feel like that's how I know I'm getting old.
You've gotten wiser.
Is that it?
It's wisdom.
Do you promise?
I promise nothing. I've gotten a lot of white hair. I have no idea how this works.
Yeah, I don't know. But yeah, depending on the day. Today, I had tea. I was actually awake this morning. So I feel like, all right, I feel pretty good. I'm just going to have a cup of tea.
All right. I'm gonna we haven't drank nearly enough this stream, so I'm gonna take a drink. But after this race.
Drove off.
Nope, keep racing. We'll do it after.
So mean. Oh my god. Okay. I'm so tired.
It's one of the harder courses because the border falls off as you hit it.
Yes. This is when we play the Nintendo 64 version of this course, my youngest child hates this course that she always tries. We follow her right off to in 7th grade. Not just to make her feel better, not usually. Oh, there we go. I think I keep going off on that corner.
5th place. Making a comeback. Oh,
there you
fifth.
go.
All right, we'll drink to fifth.
Okay, yeah, right. I'm kidding.
Cheers. Thank you for the redemption, Scuzzbod Wotus. Excuse to drink on a Wednesday night. Cheers to everyone who's joining us.
Oh.
Okay.
Yeah, somebody's telling me that Earl Grey doesn't make me old, so thank you. I appreciate it. Is that Chelsea? Of course it is. Thank you, Chelsea.
Tea Earl Grey hot. Next Generation. Star Trek Next Generation. Captain Picard. You drink Earl Grey tea. That's right. I forgot about that.
Yeah, my husband watches it.
Drink away. All right.
Somebody else. Yeah, go for it.
Just following up, Triboluminescence had a question earlier. Can Trichodesmium, I cannot pronounce this.
Desmium, yeah.
Desmium be cultured? Can we grow it in lab?
Yes, it can if you're talented. It's one of the hardest things to culture. I have actually done it reasonably successfully, more successfully than this, of course. And I think it's because I'm really a geochemist, and so tricho like to just be ignored. And a lot of people who culture tricho try and take care of them very carefully, but really tricho just want to be ignored. And so you just need to stir them once a day and then close the incubator door and leave them alone. We think that they actually have symbionts that live, well, we know that they have symbionts in the wild that live with them. There's been some really interesting work on that. But getting like what we call a pure culture or a xenic culture where they don't have any symbionts, like, I don't know that there's anybody in the, well, if there would be a very small number of people in the world who have xenic cultures of Trichodesmium right now. It's really hard to grow Trichodesmium. Oh my God. One of my colleagues here at FSU is trying to do that, Sven Kranz, and we have a project together to try and work on that. So he's attempting, and he's done that successfully before as well. So he also loves the Trichodesmium. Like I said, easy to love.
Yeah, it's the theme of the night. I like it.
And yeah, I am not the only one. I promise you, there are a lot of people who love the Trichodesmium out there.
Oh, here it is. Oh, what?
So close. Yeah.
That merits a rage.
Toad. Somebody said Toad is best, so maybe
that was Koopa.
Oh, it was Koopa, sorry. Yeah, not Toad.
That's unfortunate.
I think you're more invested in this race than I am. I got to take it more seriously. Try harder.
No pressure.
Oh, don't be sad.
All right. Neelis, beyond the element cycle you currently study, namely nitrogen, what would you be most interested in researching?
I mean, I want to see where nitrogen fixation is going on all the time and all over the ocean. So, like, is this like the if I could do anything I wanted to question?
No, this is, I think it's anything other than nitrogen. What would you study?
Oh, sorry. Why? No. So we actually are doing work on phosphorus in my lab, and we have we think we're finding some pretty interesting stuff. So we are working on phosphorus. We also have some work that we do with colleagues. I don't measure iron in my lab. I am not a trace metal chemist. You have to be not honestly specially certified, but basically you have to have lots of special training to do trace metal clean measurements from the environment. It's really hard. Especially on a boat that's made of metal, it's really hard to collect clean samples that aren't contaminated. So basically we didn't have good iron measurements from the ocean until like 20 or 30 years ago, because all the previous work that had been done was contaminated by the boat. So trace metal chemists are like a different breed of OCD. And you have to be really, really good at it. So I'm doing work with people who measure iron and are very good at it, and I'm super grateful. So I'm really interested in how all the elements sort of cycle together in this idea of sort of co-limitation and how that plays out and what it looks like in the ocean.
So what's like the dark horse element in the ocean that's not surprising, or is that surprising, like selenium or something that was unexpected?
Yeah, so cadmium actually has a distribution a lot like phosphorus, and so it can substitute in some enzymes like carbonic anhydrase So the enzyme that converts bicarbonate into CO2 for as a substrate for photosynthesis.
Right.
And then some people are interested in molybdenum, some people are interested in cobalt and cadmium. So basically all those can be metal cofactors in different enzymes that are all biogeochemically important. Vanadium on land is important in some nitrogen fixation enzymes. So those all, what else? There are a bunch of metals, but those are the ones that get a lot of more attention after iron. Iron is the monster. Iron is like Donkey Kong.
I like it. So Chelsea has redeemed Take a Drink. Perfect timing.
Oh, thank you, Chelsea.
Chelsea, cheers. Thank you for joining us tonight.
Chelsea.
Oh, Reese's Pieces jumped on it. One of the questions we like to ask, and one you were ready for. If you had unlimited funds and no ethical restrictions, what experiment would you run?
So I don't think it has ethical questions, but
you're not trying hard enough.
Yeah, you're right. You're right. I need to drink more and think about it some more. I would just want to know where nitrogen fixation is actually going on. So like count, get abundance data. So probably from DNA, and then activity data. So RNA and just map and then pair that with my geochemical measurements and try and see what they're doing, when and why. So we've got some hints of things that might be going on that people didn't recognize before, but it's hard to tie down. It's hard to really get at. So I would just be like working with the molecular ecologists and doing my isotope geochemistry with them all the time, everywhere, from everywhere, all the time. So that's the thing, is it's really hard, right? So oceanography, so I guess this gets back to some of the other questions earlier, right? So it's really hard if our processes are biologically mediated to get representative samples when we get to go out on a boat. We have to apply to NSF and funding rates, and ships are expensive, like really expensive. So you go out and you get samples in one time and place, and then you've got to try and see if you can extrapolate that elsewhere, right? And that's a hard way to go, right? And so molecular data maybe helps, but there's also these profiling floats that people are trying to put a bunch of sensors on and see what they can do, but yeah, it's hard to get the coverage that you want in space and time.
I mean, unlimited budget. Really, you have an army of measurements.
Yeah, I mean, then I'm all about nitrogen.
All right. So we could also dump like tons of, I don't know, nitrogen 15 somewhere in the ocean and seeing where it shows up.
Don't do that to me. Yeah, no. We have, and then all my measurements are toast. So that's kind of an interesting question from an ocean circulation perspective. And we kind of have done that. So again, Chem-Osh friends, students from class, right? We have done that with the nuclear tests we did in the 60s. So helium, tritium, so we've looked at that partition into the ocean through air-sea gas exchange, and then use that as what we call a transient tracer to sort of watch how long ocean circulation takes. So we don't have to dump anything. We've already done that, and people are actually doing that with atmospheric CO2 as well. So already done. We're already dumping in the weird stuff in the atmosphere and ocean.
All right. Neelis has redeemed Take a Drink. So cheers again. Thank you for joining us. Scuzzbot, thank you for the gift sub to Chelsea. Chelsea, thank you for joining us again tonight. It's a pleasure to have you asking questions. Cheers. Oh, so we're doing it again. This is Mushroom Cup again?
I guess so. Yeah, that's what happened.
All right, so we should probably do a game-related prediction. So will Angie win this round? So you took second place last time behind Koopa, and so you're replaying it again. Will Angie... We're going to do a 2-minute prediction period. So what is it? It's 5 races for an entire round? Something like that?
I I have no idea.
All right, we're doing a game-related prediction. Taking a break from science. Will Angie win this round? Not just this race, but all 5 rounds. Will she take home the gold? She took 2nd place, which isn't bad considering she hasn't really played this game at all before. You typically played Nintendo 64 version of this. Now you're playing the Super Nintendo version. Will Angie win this round? Yes or no? We need some believers in chat. Minute and 30 seconds left.
Not if I hit the thing. So I think the real question is, will I actually successfully pick up any of the sharks that they leave on the track?
So have you been using the weapons?
A little bit. I think I launched a shell that actually hit somebody once, but that one's not going to maybe. But hopefully it doesn't hit me. Yeah, sorry guys.
Well, no, you get to retry this, so it doesn't really matter. I think you get 3 retries per round. Okay. Will Angie win this round? As long as you get first here, you're still on track. We got a fair share of believers and skeptics with this one.
Oh, that's fine. I'd be skeptical, but see.
Shiloh is all in on win. No pressure.
Shiloh, that's so great. Thank you.
Your support is appreciated.
Yeah, that's lovely.
All right. While the prediction's waiting for an answer, we have several more rounds to go. Any questions? Anyone just joining us? Is Ask a Scientist Gaming. Mediocre gameplay, except this time it's going to be awesome. With expert level science, our guest today is oceanographer Dr. Angie Knapp, who studies marine nitrogen cycle. Really cares about ocean and nitrogen and where that goes, as well as its impact on things like CO2 fixation. So if you have any questions, throw them in chat right now and we are happy to answer them. All right, Ninja Physics, have you observed any ocean atmosphere coupling due to phytoplankton activities? How does phytoplankton affect the atmosphere?
Good question. I don't study that so much myself, but one thing that's interesting is phytoplankton release a molecule called dimethyl sulfide, that is a cloud seeding agent. And so there are certain kinds of phytoplankton, especially coccoliths, that release this molecule that we think see, if you get me talking science, I can't drive in.
Oh no.
It's so rough. Sorry, guys. So certain phytoplankton that are sort of endemic to certain regions of the ocean release these molecules called dimethyl sulfide. So there are definitely people who are trying to look at those links between the ocean and the atmosphere. Actually, sort of a different way around. Those dust storms that we get in the summer, especially if you're like in the Tampa area or St. Pete. So that's African dust. And we get some in the Gulf of Mexico and on the West Florida shelf. And we actually think some people have suggested that it stimulates trichodesmium. It's all going to come back to trichodesmium. And so that's an ocean atmosphere coupling that we care about is, we need to know where they get their iron from. And on the West Florida shelf, it may be atmospheric dust during the summer, but we also think there's some other sources that are underappreciated.
Ninja Physics follow-up, so they burp clouds into existence.
Yes, exactly. That's the nice way of saying it. You could say they do something else, yes.
You mean they fart clouds into existence? Yes. Is that... we're an 18-plus stream. You can say it.
I can say that, yes. But they're phytoplankton. They don't have a digestive tract. So.
I see.
See, I got Donkey Kong with the red shell.
Nice.
He deserved it.
All right, casual question from Chelsea. What's your favorite book for pleasure reading, not a textbook?
Chelsea, such a good question. I love Nicole Krauss, and I loved her The History of Love. I love The Tiger's Wife by Thea Obrecht. I think those are great. So those are for fun. Also, should reread one of those. Oh,
Came back with a win.
good questions from Chelsea, pushing me over the finish line.
All right, believers, there is hope. There is still hope. Pull out the wins. Well done. All right, Wotus wants to know, what is your favorite formula/equation?
So I am going to go with mass balance equations, because they cannot be violated, right? You have to abide mass, and we use isotopic balances all the time, right?
So elaborate what mass balance is.
Yeah. So that means we care about, when I'm saying we try and understand what fuels phytoplankton growth, we maybe are looking in a certain place over, say, the course of a year or something like that. There are a couple of time series stations in the ocean, so at Hawaii and Bermuda, that have been funded by NSF, where people go out monthly, and so there's these time series stations, and we just have these invaluable records where we just don't have that in many places of the ocean. But so we can evaluate, we can ask questions like what happens over the course of a year at these places. And so we often ask, what are the quantitatively important sources of nitrogen fueling phytoplankton growth on an annual basis? Let's say Bermuda. And I try and answer that question using stable isotopes, because we can try and look at what comes out of, say, the euphotic zone, so the upper 100 meters where sunlight reaches, where sunlight reaches and phytoplankton grow. And we can collect the organic matter that sinks out of that euphotic zone and measure its isotopic composition, and that provides a constraint on the sources of nitrogen that are fueling that phytoplankton growth, right? So in steady state, what goes in has to be equal to what comes out, right? What goes up has to be equal to what comes down, and they have to balance in terms of the amount of nitrogen that goes in has to come out. Otherwise, it has to accumulate in a pool, right? So if we're in steady state, things just have to balance. And that's what I mean by that. And so I do a lot of isotopic budgets that help us identify when and where nitrogen fixation is important. And it's never as important as I want it to be. We have found it being important recently in the Southwest Pacific. That's also a pretty cool story. But did you see that?
That's 2 first place in a row.
That was amazing. Okay.
Cheers to that. Clip it. That's amazing.
That was, right? I deployed that shell right at the end.
Chelsea has a follow-up.
This is just dumb luck. This is not skill, I promise.
I think we're reaching skill here. This is several in a row. We have several data points. All right. Chelsea wants to know, is the nitrogen cycle balanced yet?
It depends who you ask in the day, Chelsea. Currently, estimates suggest that the ocean is losing nitrogen maybe 2 or 3 times faster than it's gaining.
Losing it to?
By denitrification, which we don't really believe. So current estimates, measurements suggest that rates of denitrification are 2 or 3 times faster than rates of nitrogen fixation, which makes no sense, right? So Chelsea's going to get me to just monologue here. So she's heard this in class. So if that were true, then the ocean would run out of nitrogen in something like 1/3 of its residence time in the ocean, so like 1,000 or a couple thousand years. And then carbon fixation would come to a screeching halt. There'd be no more plants. It would be a big problem, but we don't think that's happening in the ocean, even in the relatively recent past, based on the geologic record. And so that means that we've either overestimated rates of nitrogen loss, like denitrification, and or we have underestimated N2 fixation. So that's one of the reasons I study nitrogen fixation. So we're trying to see if we've underestimated it and we maybe haven't figured out where it's going on or what. And so we're still trying to identify who are the dominant nitrogen fixers in the ocean, where they live, when, where, why. This is why I want to measure the whole ocean for nitrogen.
So that's interesting. So you have like a dark matter-like hole in nitrogen fixation right now.
Well,
that's not
perceived.
Fully understood.
Yeah. Perceived. And most people don't, it can't really be that out of balance, right? Geochemists who take the long view are like, that's just not plausible, right? Or it's not consistent with all these other measurements and our understanding of the system. So, but it does mean that we don't understand it. And again, because this is biology that's at work, and biology is kind of fussy and very sensitive. They all have very different sensitivities. It just means we don't understand it, right? What controls their distribution very well. So, yeah, this is why we work with biologists, and we have to look at all the elements that can mimic photosynthesis and NT fixation. So iron and phosphorus and all these things, and trying to figure out what's going on, right?
I like it. So with that 4th place, you squeezed out another one. So you're still holding strong here, believers. Have faith, have hope. Still gonna pull out the win here. We'll do some easier, more casual questions while you're playing.
Yeah, seriously. Another way.
Let's start with the most memorable, impactful moment of your scientific career.
It's kind of tough. One thing that was cool was when I was in college, my college invited Lynn Margulis to speak, and I got to have dinner with her, which was pretty awesome. So she is part of the one who coined the symbiotic theory of evolution, right? So that, there's sort of a ribosome and other things, and one engulfed microbes engulfing each other to form eukaryotic life, right? And so I just think that's amazing and super interesting. And so, I talked with her and thought about doing grad school with her, and she accepted me to work with her, which would have been awesome. But then she was like, well, she was at UMass Amherst, and she said, you got accepted to some other schools that have better resources. You should go and work with them. And I didn't really understand that at the time, and I thought that was kind of a weird thing to say. But I listened to her, and it's really good, really good. So that was also very generous of her.
So in retrospect, that made perfect sense then.
Yeah. Oh, a banana peel cost me. Yeah, so generous scientist out there. Yoshi just stuck his tongue out at Mario, I think. That was funny. Donkey Kong.
Oh, interesting. Ninja Physics 44. Have you faced challenges as a woman in STEM?
I mean, sure.
I mean, there's passive challenges and then there's active challenges.
Yeah.
I mean, do you have any crazy stories that you want to share?
Not that I want to share.
That's fair. I mean, so I guess follow up on that. So physics and math are dominated by males. Biology is swinging more females than males. Chemistry is somewhere in between those two extremes. How is oceanography? Is that
I got
male?
one. I think we have 25 to 30% female faculty in my department.
Oh, that's really good.
So
actually.
It's a lot like chemistry and yeah. So but I think we share the same challenges that a lot of departments share. It's just it'll be a long, slow road.
So you're winning by 4 points. Is there one more?
It's amazing.
Is this the last race?
All right. It says final. Is that what that means?
I think it is. This is the final race.
All right. No pressure.
Chelsea, we talked about lab disasters. What's your most comical lab disaster?
Oh, comical lab. Oh, okay. Yeah. So I told you guys we measure the nitrogen isotopes in my lab using microbes, right? And this is funny in retrospect, but definitely not at the time. So on my first sole PI NSF grant, I had some colleagues collecting samples for me. And one way that people used to preserve nutrient samples to prevent microbial alteration was they would poison them with mercuric chloride to kill anything that was alive in there and prevent that from changing nutrient concentrations. That's great. So some people still do that. But that's a bummer if the way you measure nutrients is using microbes, or the isotopic composition is with microbes. And so they sent me samples that were poisoned. And I tried to measure the isotopic composition. And I was pretty upset. I was pretty pissed. Yeah, so I still work with these colleagues, but I learned my lesson: never tie their samples to NSF dollars, because that was super stressful, especially pre-tenure. So we were actually able to pull it out and still measure the isotopic composition we had to increase the number of bacteria doing the analysis relative to the sample volumes. We minimized the sample volume and increased the number of bacteria so that basically there were enough enzymes that could carry out the analysis before the bacteria died. So we were able to salvage the samples, but it was not fun. And I don't recommend it. So, and then what was funny is they denied it, but we had colleagues in the department who could measure.
Oh, is that it? Did you take out the win?
Measure? I don't know, man. But yeah, so there was totally mercury in samples. They said they didn't measure. So funny-ish now. Definitely not funny.
I could just imagine the rage that night. Like, that's just go take a shot or two. That's
actually, it was funny because I was on my way to a conference in Hawaii, and I was reading through their because I was trying to figure out what's going on with my samples, and I was reading through their protocols, and I had an aha moment. Like, I saw that they poisoned them with mercury, and I was like, oh my God, they killed my samples.
Oh. So prediction. The believers win. Will Angie win this round? The answer is yes. There is her gold trophy. She has won. Believers, you take the win. It's about a 50-50 match in believers and unbelievers. So, congratulations. Let me choose the outcome here. Will she win? Yes. Congratulations to who got their internet points. Shiloh going all in. Well done.
Thank you, Shiloh. I appreciate that.
You should move on to the... can't you do a different cup?
Probably. Okay. How do I back out?
So go to no.
Oh, sorry, Ken. How do I back out?
You could just reset. It's probably the easiest way.
Okay, this is in... Yeah, here we go.
It paid off. Reese's Pieces has used the points to redeem Take a Drink. Angie, thank you for stopping by, playing Mario Kart, talking science, and drinking with us.
Thanks for coming, you guys.
And
I'll Trico all the time.
It's what we do here. I'm going to have to come up with a Trico emote. I don't know what that would look like, but if you have any ideas.
It's cute. Yeah, it's,
it is.
Get it wrong.
That's awesome.
It's they're like bow ties. Wait, Ken, I did it wrong. Okay.
One player game.
Go back. Yeah. Okay. Go to time trial.
Mario Kart GP. Hit that one. Mm-hmm. Can you pick the race 50cc?
I think I did the 50cc before. So will this give me different courses?
I don't think so. I think this one will give you faster. I think you pick it later. I might have screwed up. So go no.
Oh, okay.
I would stay on 50cc.
Yeah.
Go up 50. Click that.
Yeah, I think this is it, my friend. This is just what we get with Super Mario.
I think on the next one you get to pick between flower, mushroom, and star cup. So I'd go to flower. Oh. Yep, go down to flower.
Thank God.
You got your gold on mushroom. There we go.
Thank you. All right. Thank God, some different courses here.
All right. Easy question. Favorite meal, favorite finger food. We'll start with favorite meal.
Favorite meal? What a good question. I don't even remember anymore. I have kids, you guys. I don't know what my favorite food is. Whatever they will eat.
Whatever they will eat is the right answer.
How many meals do I have to cook for dinner? Anything with goat cheese. You know? Anything with goat cheese.
Goat cheese is the answer. Favorite finger food, also goat cheese.
Yeah, cheese and crackers. Yeah, like my perfect meal is sort of like cheese and crackers and wine or something like, you know, and like nuts and dried fruit or something, fresh fruit or something like that. So, yeah.
Do your kids like goat cheese? Probably not.
No. The 12-year-old is getting more adventurous, but 7-year-old has a palate like my younger twin brother's, which to this day is still like peanut butter and jelly and like plain cheeseburgers and pasta with marinara sauce. So yeah.
All right, Asimov wants to know, how do you keep your samples then without mercury?
Yeah, good question. So we filter and freeze them. So filtering keeps the microbes out, and then we freeze them at -20, so not -80 like molecular samples or the Pfizer vaccine, but just regular freezer temperatures. And that, so we freeze and thaw our samples. We have to thaw them to analyze them, and so we have a lot of practice freezing and thawing, and we don't see nutrient concentrations or isotopic compositions changing as a result of all that freezing and thawing. So that seems to be okay for us. So we often do like 0.2 micron filtering or to 0.7.
Nice, saved the third place. OpenSet has a comment. I was just reading about Dr. Knapp's research focus. Reminds me a bit of the research another science streamer here called Diatom's Attack.
Oh.
I'm not familiar, but we should follow Diatom's Attack.
Yeah, I want to know who Diatom's Attack is.
Yeah, and what do they play? Or do they just stream science?
Right. Exactly. These are the important questions.
Diatom's Attack. Sorry, I'm going to look this up quick.
Yeah. All right, I'm going to try and stay on the course this time and not fall off. That's my goal.
All right, so there's another science streamer, Diatom's Attack. If anyone's interested, does marine biology related things. Looks like a bunch of video clips of scanning electron microscopes of different diatoms.
Diatoms are so beautiful. Yeah, you should share some pictures that they have because those are beautiful.
So he is at Indiana State and looks at diatoms from East Africa Rift and Great Lakes.
Oh,
That's
so
fun.
Freshwater, not ocean.
There's a surprisingly large body of science streamers on Twitch. They typically don't play video games while doing it, so we have our unique niche here. But yeah, it's worth checking out. There's a lot of computer programmers, but there's also a lot of science streams available. So OpenSet, thank you for that link. You guys follow them if you need to.
Stay on. Oh no.
Diatom's Attack. He's a paleoecologist.
Yeah, sounds like they do terrestrial diatoms, but yeah, sort of reconstruct. Probably like we were talking about, reconstructing how temperatures have changed and
Rage. All right, we'll build upon the rage. What's your biggest pet peeve in your field? This could be something people get wrong in literature or reviewer related or anything.
Sample collection. Okay, so this actually should be like a quiz question. So friends, and I know Chelsea knows the answer to this, she's heard me complain about it. What happens to water when you freeze it? It expands. So if you go to sea and you collect samples for me and I give you bottles, should you fill them to the top before you put them in the freezer? No, you should not.
This is a systemic problem.
Yes, you have no idea how many fancy PhDs, like full faculty members, they forget about this when they go to sea. And you fill a bottle all the way to the top, and you have no room. And then the sample's ruined
amazing.
because salts freeze last, and nitrate is salt, and the organic matter also freezes last, and it gets extruded out the top. So we can tell if you've done this because it's a Nalgene bottle that we send. It's a tiny one, but it has that threaded cap, and it crunches. You can hear the salts in there that were extruded when it was trying to freeze. So we know if you overfilled our sample, and then it's all messed up. Like the nutrient concentration profiles are all jacked up. They don't look right. And I can tell. If you did it, I can tell. Oh no, I'll know. So don't try and even pretend, because I'll know.
I mean, that's interesting.
I'll drive off the side, but I'll know.
So the freezing in that case, it's doing a gradient purification to kick out the stuff you want to measure.
Yes.
That's how they purify silicon for solar cells and things like that.
Yeah.
So,
no, we recrystallize some of our reagents to purify them. So yeah, we do. I do this for some of my stuff. Yeah. So yeah, jerks. Yeah. So you get nice pure water, but yeah, it doesn't have my analyte in it anymore, or not the right amount. So that's a pitch.
Oh man! So you have to have a talk through with your students and with your collaborators.
I should show you. I have made a graphic that spans languages, because I work with collaborators from all over. So this might tell you something about who's been doing this to my samples. So I have it, and it is language free, and it's I draw a happy face on the bottle and like a fill line. I had an undergrad, was it last summer or the summer before? He was labeling bottles for me, and I had him in the language of my colleagues who had maybe overfilled this, who maybe also were the same colleagues who poisoned my samples with mercury. I had him write in a different language. Fill line, stop here, do not fill above. And he wrote that on every single bottle for like hundreds of bottles. He drew a line, fill here at the shoulder of the bottle, stop, leave space to expand. And so there's like a graphic that I have. I can pull it up on my computer. Like happy bottle, space for freezing and expanding. Sad bottle, one that has water coming out when it freezes.
Man,
that's to the inside of all the stuff that I send out. So no one will ever care as much about your samples as you. So if you send bottles for other people to collect samples, it's like, don't get too attached to it. Like it'll be nice if it works out, but you have to be prepared for those samples not to work. So if you really, really want a sample, you have to be on the boat collecting them yourself. Or one of your grad students.
That is one of those, like, you could have a brilliant scientist that just a common sense moment, like if you've ever frozen breast milk, like these rules and stuff. Like it's man, that's wow. Yeah,
well, and being at sea also, like it's hard, and most people, like I said, are on seasickness medication, which does mess with you, like honestly. And like you're just really busy and trying to manage a lot of stuff, and nobody, like you're working 24 hours a day, and so it's pretty high pressure. Like you're working around the clock out there, and
with no depth perception.
For some people, yeah. And, so there's a lot going on, and if they're not your samples, you're just not thinking about it. You don't care. Care. And so, like I said, no one will ever care as much about your samples as you do. So, some of my colleagues, I have a lot of sample collection going on in the South Pacific with ports in like Tahiti and New Zealand and New Caledonia. I have never been to any of these places, but basically I need to be going to collect these samples from I can't do this, of course. This is really brutal.
And you're in rage mode right now. It's okay.
Yeah, I'm really irritated about mercury poisoning and like people overfilling my sample bottles. All my colleagues have heard me complain about this. Like, oh my God.
I mean, that's awesome. You just had a straightforward answer to that question that I did not expect. I appreciate it. I'm very sorry you have to deal with that.
Basic science principles, right?
Yeah, no,
like
it is.
Everybody
It really
here
is.
can understand. So yeah, it's one of the reasons why life on this planet is possible, is because water in the solid form is less dense than in the liquid form. It's totally that's bananas. That's bananas, right? Like that should not happen. It's totally bananas, but it's true.
Fun factoid, that's why we'll never bring Walt Disney's head back to life. Anytime you freeze a head under normal conditions, it will shred every cell in it. There's flash freezing you can do. That doesn't do crystallization, but that didn't exist 50 years ago when they froze his head. So, if that's true at all.
I didn't know this about Walt Disney, but okay.
Yeah, that's the rumor. I don't know how real any of that is, but yeah, the freezing technology back in the day was terrible. So they were selling you snake oil to freeze your head and just shred every cell in your head. So,
it absolutely would. Yeah, rip it right open.
That's awesome. Is there any pet peeves with literature? I mean, is there like experimental details that people don't give that are?
Yeah, all the time. So I think There's also sort of a cultural difference. Like I said, I sort of consider myself a geochemist, and then there's sort of the biological oceanographers, and we sort of come at things from very different perspectives. We sort of want different things from the literature or experiments and controls or not controls. Let's do controls. Time zeros. Oh, so this is a good one. This is a really good one. So people who do nitrogen fixation rate measurements, they'll add 15N2 labeled gas, and then for 24 hours to a water sample, and then after 24 hours, filter it and see how much of that 15N has made it into the particulate biomass. So that should be nitrogen fixer biomass. People didn't do time zeros. And so they assumed an initial isotopic composition for their time zero, because, you know, the way you figure out the rate of N2 fixations, you have time on the x-axis at 24 hours, and then the change in the isotopic composition on the y-axis. But if you don't constrain your time zero, you can make it do whatever you want. Yeah, exactly. And that's kind
awesome.
of what happened, actually. And so to be fair, people thought it didn't matter, and it was always kind of one isotopic composition of sort of particulate matter. It was sort of constant, but it's not. Their stuff was always done in the Atlantic and in the tropical Atlantic. It's a certain value there, but it's very different in different parts of the world's oceans. And people weren't taking that into account, and that led to some pretty problematic N2 fixation rate estimates. And so that actually sort of damages the credibility of the entire field when that happens, right?
And so you get sent papers to review, and you have to tank them based on that?
Oh yeah, for sure. So we just had a methods. So myself and a bunch of colleagues actually had a workshop a couple years ago and sort of tried to outline best practices for how to make nitrogen fixation rate measurements in the field. And so we talk about that and a bunch of other problems with the measurement. And so that paper just came out last year.
I mean, that's crazy. Like it's an entire paper. That's months and months of work and sample collection and thousands and thousands of dollars that are just gone because of that.
Well, people published it though. Like this uses what used to just get published and was okay, and people wouldn't say anything. And so that's sort of like when I say the biologist's point of view, like they didn't think about it. But then like geochemists sort of caught on and were like, hey, wait a second. If you actually think about that, like you're actually really inflating the rates here. So, I think that's just sort of a natural evolution, right? People just sort of pay attention more over time, whatnot. But But geochemists sort of view things in one way, and the biological oceanographers sort of view things in another. Sometimes gets a little heated.
Yeah, so this relates to Reese's Pieces question. Any culture clashes with other scientists from other fields/countries?
Not really countries. I don't think there's sort of a countrywide division, but there's definitely a biological oceanographer versus chemical oceanographer division. So you're either a chemical oceanographer / geochemist, or you're a biological oceanographer, and they sort of have different worldviews and perspectives on how you run the experiments. And so I feel like everybody has important things to contribute, and best practices have to be in the middle, right? They have to take into account everybody's concerns. And so I do try and work with both sets, but then depending on the individual, some are more willing to hear
That's
Isn't
fair.
that always true, though?
Yeah, exactly. All right. Chelsea wants to know, when you were a kid, what did you want to be when you grew up?
I had no idea, but I did not think I was going to be a scientist. I don't know what I thought I was going to be. I didn't really have an idea. I think when middle school, I thought I was going to be a fashion designer, which makes no sense because I just want to wear jeans and a t-shirt all day long, forever. So I don't.
See, that was your hours of watching Gem and the holograms, right?
Yeah, probably. That's probably it.
Indoctrination.
Yeah. Exactly. So I didn't have a good sense. I definitely did not think I was going to be a scientist, though. I think my mom said I was at some point, and I was like, she knows that. Oh God, my mom. And so she said either that or a lawyer.
You are going the wrong way.
Oh, again. I hate that.
Yeah,
guys, I don't think I'm going to win this round.
That's okay. No one has imaginary internet points on it, so no stress.
Okay.
We're in the partially drunk phase of the night, so it's okay.
Honestly, it happens when I'm sober in Mario World.
That's fair.
Nintendo 64. It's just sometimes.
ZP Bruins 1233, a spammer has a question. Want to become famous? Buy followers and primes and viewers on. Can we ban that person, please? Come on, thank you.
Will it get my next NSF grant funding?
OpenSet has a very important point. We're already famous. Thank you.
Fabulous.
We're not Twitch famous, but we're already famous.
Okay, excellent.
Man, I wonder how often that works. I mean, presumably enough to keep doing it. It's the Nigerian prince scheme of Twitch viewing, which is kind of crazy. What they don't understand is, I want to clarify for everyone, we are not in this for the viewers and the money. We just love the science and we want to share it with as many people as possible. This is an awesome medium to do that while playing video games and drinking. So thank you all for joining us. Yeah, should we write that into our NSF grant? I need broader impacts funds to pay ZP Bruins 1223 to get us viewers.
No money went to them. Yeah.
That's amazing. All right. So anyone just joining us, this Ask a Scientist Gaming, mediocre gameplay, expert level science. Our guest is Dr. Angela Knapp. She's an oceanographer that studies the nitrogen cycle. We've learned a lot about nitrogen fixation, phytoplankton, evaporation, CO2 processing, a whole lot of different things tonight. It's been a lot of fun. She also won the Mushroom Cup, I want to remind everyone. So, believers, kudos to you for getting your internet points through that bet.
Even though I'm in last place right now.
That's fine. This is a different race. So,
it's muddy.
If anyone has questions of any kind, please throw them in chat. You have to believe. Have faith in our scientists. We're going to do it. We'll pull out the win.
I'm trying.
Man, that's crazy. They just go from stream to stream and just spam with an advertisement, and then presumably we banned them and reported them. Thank you, Wotus, for the ban and the report. But that's their job. They just go to streams and drop advertisements in. It's kind of crazy. Yeah. But so it goes.
Reminds you that even on Science's hard days, could be worse. Could be doing that. Could be spamming, Ask a Scientist Gaming, don't you?
Yeah, sometimes, I'm angry about our job, but our job is awesome. What we get to do on an everyday basis is amazing.
Yeah, we have a lot of freedom, and for Pete's sake, I get to write grants and try and get a boat that the U.S. government pays for, and then drive it around where I want and collect whatever samples I want and bring them back to the lab and measure them.
But only fill the bottles partially to the lid.
Oh my God! Otherwise, you're throwing away the NSF money, and you're going to end me. So, and then don't put mercury in them. Thank you very much. It's also for boat. That's not a modern practice. That's from the 1960s. So. Some people still do it, though. This may be where we get down to different countries. There are some countries where they still do that.
All right, so we're going to do our final science prediction of the night. So those of you that have your internet points, we're going to spend them now. If you've been in the stream, just click the follow button. If for one reason for the points, for another reason, just get us, number of viewers, and that helps us with visibility. But the question is, so oxygen concentration has changed over the course of the history of the Earth. Was there more oxygen 1 billion years ago or now? Is it higher now or lower now than it was 1 billion years ago?
Chelsea, this is for you if you're still out there.
Shout out to Chelsea. You better get this one right. Is it higher oxygen content now or higher 1 billion years ago? Higher now or lower now is the question.
Oh, I should just end it. God, why do I keep going back to this? It's brutal.
We can go back to mushroom cups. We don't have to keep playing.
It's okay.
All right, you guys have a minute. Is the oxygen
for
content
punishment here.
higher now or a billion years ago?
Are these Venus flytrap plants that are on the course? What are they? I just keep crashing into them and they're awful. These plants in the mud.
All right, 30 seconds. Oxygen content now versus a billion years ago. Is it higher now or is it lower now than it was a billion years ago on Earth? A billion years ago. That is... So I'm a spectroscopist, and I do time-resolved emission, and I do femtosecond measurements. So 10 to the minus 15 seconds. And then you pull up a word like billion. That's, 10 to the ninth the other way. It just...
It's hard to think about.
Yeah, work on very different domains.
Yeah. My work is mostly in the modern, but I do hang out with some people who do paleo stuff, and it's pretty awesome. I really enjoy the paleo stuff. So Chelsea is one of those people. I don't think she quite goes back a billion years, but she definitely appreciates the billion-year-old stuff.
I like it. All right, betting is closed. It looks like we have about a 50-50, a 3-2 higher now versus lower now. Earth 1 billion years ago versus now. The answer is it's higher now.
For sure. So Earth is not what we would call a hospitable place when it first evolved, and so it evolved like 4.6 billion years ago, but still, for the first 3.5-4 billion years, Earth didn't have enough oxygen to support macrofauna, right? Or for the most part anyways, maybe about 500 million to 1 billion years ago, you could get some in some parts, right? But not on land. And so, yeah, Earth was a very, very different place. And so in one of my classes, I've taught Habitable Planet. We sort of talk about, and in History of Earth Systems, we talk about how the atmosphere of the Earth has evolved over time. And most of the oxygen today comes from photosynthesis, but oxygenic photosynthesis was not the first metabolism to evolve. And in fact, it was probably other kinds of photosynthesis that evolved first before oxygenic photosynthesis. So like in the green and purple sulfur bacteria. So they would have evolved sulfur, like sulfate or elemental sulfur instead of oxygen as their waste product. So totally wild, but yeah, we'd not be able to breathe. It would not be a fun place.
So congratulations, everyone that said it's higher now than it was a billion years ago. I'm going to pop up a graph. I'm going to put this over the screen.
Lovely.
So there's the Wikipedia graph. Sorry. Let's get that there. So it's not always been higher now. There was a phase 300 million years ago where we had this peak where it was higher, and then it went down to now. So that was the major growth phase in biology, is that correct?
Land plants, yeah. And then not just so it was also sort of burial of a bunch of organic matter, which also increases atmospheric oxygen concentrations.
Yeah, so there's a billion years ago, there's now, there's everything in between. There's a peak at 35%. That's a lot of oxygen.
It is, and so there have actually been a lot of modeling studies, this is kind of wild, where people try and estimate how much oxygen the atmosphere could hold before you would have spontaneous combustion. And they think that that's basically the theoretical upper limit, and that above that there would just be wildfires all the time. And so they even have charcoal layers in the rock in the geologic record from sort of that time period that they think may be consistent.
Yeah, giant bugs. Chelsea has a good point, giant bugs. So, like, we have a circulatory system, but bugs don't, right? They have to rely on diffusion, which is limited by surface area and oxygen content. So when the oxygen is higher, you get bigger bugs, which is crazy.
And, like, dragonflies that were... Chelsea, how big were those dragonflies? Like, a meter across is the wingspan or something ridiculous like that. Like, it's bananas. Again, this is where, scientifically speaking, bananas is appropriate.
Yeah, so, no, that's like a grab your cat type dragonfly. That's
yes, exactly. Florida's working on it, right? Florida's working on that level, but the backwater swamps, you might find that kind of situation. This is not my course either.
That is amazing. All right, OpenSet has a question. Do you have any good oceanography jokes?
No. I can never remember any kind of joke whatsoever. So yeah, no, I have no jokes. I'm sorry.
But they do exist. You've heard them before. Some are funny.
No, I can't say. Not even at sea. Not even from like their ship's crew or anything. Maybe I wasn't hanging out with the right people, or they just weren't telling me the jokes. I don't know. But
does anyone in chat have a good oceanography joke?
There must be some out there. I just don't. I'm not the person who has them.
Oh no, Diddly Doo has one.
Oh, no.
Why do algae couple tend to drift apart?
Why?
Because they prefer planktonic relationships.
Oh my god. Okay, that's good. Okay, all right, I like that.
Diddly Doo, you have approval from Dr. Angela Knapp for your oceanography joke. She will forget the joke, but right now it's amazing.
It's not personal. It's just not. My brain cells are all taken.
I'm sorry, my delivery did not do justice to that joke, but well done. It'll be on YouTube. Check out our YouTube channel.
I love it. Maybe it'll work its way into one of my classes. I'll try.
Diddly Doo. That's amazing. We asked for an oceanography joke and chat delivered.
I know. That's
I love
huge.
Twitch.
I really appreciate that.
Yeah, no, this is why I love Twitch. Diversity of thought and expertise and knowledge backgrounds comes together.
Yeah, I can't promise. Yeah, I definitely don't have the jokes. That's not my skill set.
That's amazing. Oh, so close.
Yeah.
Oh, man. Unrelated fun factoid, I got my vaccine shot for COVID yesterday.
Nice.
Just the
Where'd
first
you
one.
go? What did you get?
Publix, and it's the Moderna one.
Nice.
So I have a follow-up on April 26th.
Okay. My husband and I drove to Alabama a couple weeks ago, so we are scheduled for our next shot. He goes next Monday, and I'm going to go back to the mushroom. Next Monday, he's Monday, and I'm Tuesday. We're trying to get scheduled. We have the Pfizer. We're trying to get the Pfizer in town, but we haven't been able to get it in town. So
that's interesting.
We're gonna keep trying. Yeah, I think FSU's like. We'll do first shot, but if you got your first shot somewhere else, you have to go back where you got it from. And
that's
CVS
interesting.
CVS, I almost got a second shot at CVS, and then it got taken while I was in the process. So I think supplies a little tight right now. Not surprising. But we can get it. So that's okay.
I saw a statistic that the U.S. is at 15% full vaccination and like 25 the first shot or both shots, which is amazing. Like our numbers are turning around really quickly.
I know.
We are on the brink of in-person Ask a Scientist gaming, so
that'd be great.
I want to give a shout out to Angie right now. We troubleshot the shit out of making this happen. We went through 3 different computers, 3 different operating systems to try to get this stream to work, because it's hard to share video game video across platforms and whatnot. So Angie, thank you for spending the time and effort, because it's awesome to have you as a guest.
Well, this is fun, and we got to give my husband some credit too. I joke that I married for tech support in addition to love, and he's definitely.
Cheers to him. I'll drink again.
Exactly.
Kevin, is that his name?
Kevin, yeah, he's
Kevin.
In physics.
Cheers to you.
So you can get an astrophysicist on. Actually, you already do. He works with David Collins. So
Hey, if he wants to be on, let's do it.
There you go.
Awesome. Oh man, OpenSet has another joke. Are you ready for this?
I'm ready.
Brace yourself. Do you know why so many people on boats drink?
Why?
Peer pressure.
Oh my God. Okay, yeah.
You had to see the spelling.
Yeah, I get it. So So American boats are dry. They're very strict policy. But European boats, South African boats, not dry. And I have not been on a French or a European or a South African boat, but I have sent my graduate students in the past few years on them. And apparently the South Africans have the world's nicest and newest icebreaker. And apparently they have white linen table service at dinner, and like you have to wear a collared shirt, which is ridiculous because when you go to sea you are wearing your dirtiest, grubbiest clothes. Like we say cruise, but it's not a cruise, right? Like you're getting engine grease and salt water on you, and your hands are raw, and you don't sleep, and like you're just disgusting. It's like camping, forever. And so,
they do white tie only, like black tie only. That's crazy.
Exactly. And so I'm like, what are you talking about? I have not had this experience. So yeah, I need to be having the port calls in Tahiti and having wine with my dinner at sea. I think, this needs to happen in my life.
So how has your research been affected by the COVID? I mean, in terms of restricting travel and whatnot.
It's been hard both for getting, work done in the lab, like it has for everybody. Mm-hmm. And then, travel. Yeah, we had cruises. So the poor people at NSF who do ship scheduling, it's just been a nightmare. So, ships are really tightly scheduled and people have to go at certain times of the year and whatever. And so the whole ship schedule has just been thrown upside down.
So pause. My familiarity with the NSF is they give you money and then stuff happens. But this sounds like they have a mechanism where they control the boats. How does that work?
There's research vessels, and so it's actually owned by the Navy, but it's administered through NSF, I think maybe in coordination with the Navy. But so I say I want to go to the North Pacific, and I need to go in May, and I need to be out there for 28 days. So I request ship time in my NSF grant, and that ends up being expensive. But the way NSF Oceanography runs it, they don't factor in the cost of ship time into your total grant proposal, right? So we make a budget for our grants that includes faculty salary, grad student salary, supplies, travel, things like that. But for NSF, we don't have to factor in the cost of the ship time, although they look at that cost. They have to be able to afford all the ship time. But then, from just a logistical standpoint, people are like, well, I need to go to the North Pacific in May, but this other person needs to go to the South Atlantic in October, and there are only 4 boats that, there's basically one boat in each basin that they call global class that can go for like a month or more. You really can't go more than like 6 weeks without coming back to port.
Ooh, sniped by a shell. That was brutal.
I know, it was rude. So yeah, just the whole ship schedule has been a mess. So there are cruises that were supposed to happen last January that are hopefully happening. It's January 22. There are all kinds of, so the whole ship schedule, like everybody's science is just kind of, guys.
Do you
right into it.
So do you have I'm just curious. I don't have any idea what this costs. Like if you want to do one person for 28 days at sea, like what is the rough cost of something like that?
So the way we think about it is not like how much it costs to send a grad student, but it's how much to take that boat out and the crew that goes with it. And so like the global class boats that can go the furthest out into the very middle of the ocean and have the longest duration, like 3 to 6 weeks or something like that, I think they're probably $35,000 or $40,000 a day.
Wow. That's crazy. Yeah. So you just write that into the NSF grant and they do the math afterwards.
Yeah, I just say I want that.
Wow.
And then if my proposal gets funded based on the science, they give it to me. Which is why it makes sense to write in collaborations with people in other countries who have ship time. Yeah. Even if they poison your samples or fill your sample bottles.
The mercury's worth it.
Maybe. I don't know.
$35,000 mercury. That's amazing.
Yeah.
Well, Neela says
yeah.
redeemed. Take a drink. We'll drink to that.
Yeah.
Saving money, poisoning samples. Well, welcome to the life of an oceanographer.
Trade-offs, my friends, trade-offs. And so, yeah. And that's not even like, some people have like the Alvin where they go in the submersible, or they do ocean drilling or something like that. So those are even more expensive. So,
I mean, so how real are those numbers? So I have a colleague that does like nuclear chemistry, and he shows me like a 10 milligram sample of Einsteinium, and he says, this is $1 million sample. But like at that point, you can't even estimate real numbers, but it's tangible, like the cost of personnel.
This is absolutely
real.
Yeah, for real. And so they even tell you, so I have to do a ship time request when I'm requesting ship time in addition. So I submit that with my other NSF documents, and it tells you how much it's going to cost for the class of boat. So for the global class, I think it's 35 or 40K a day, and usually you're out for 21 or 28 or 35 days on one of those. So it's like $1 million.
That's crazy.
Very real cost. Yeah. And so that's a huge fraction of the NSF oceanography budget, right? Is the ship time. And so that, limits how many people they can fund each round. And so then, they've already committed to all this science, right? And getting back to COVID and the logistical nightmare that is this, right? So then trying to rebalance all the grants that have already been funded with ship time that was already committed, that is region-specific, times of year-specific, and then trying to redo it is really hard. So there have been some cruises that have gone out, but what they've done is they've required everybody to quarantine for 2 weeks in a hotel on site beforehand, and NSF is paid for that.
Wow.
Which is wild. And they do COVID testing before they fly there, and then after they're quarantined, or before they go into quarantine. And then, and then they've made rules like they've only let cruises go out that are within, like they can easily get back to land if people do come down with COVID right after they leave for whatever reason.
Oh, wow. So COVID that's a red flag. That's an ending event. Everyone comes back.
So yeah, I haven't been involved in too many discussions about this, or it hasn't impacted any of my personal projects, but the stories that I hear, different institutions, different countries are doing it differently. So I did send a grad student on a French cruise, and their rules were a little bit different, and it was pretty wild. So it was in January, and it was in the island of Reunion, which is in the Indian Ocean, like in the southern Indian Ocean. Really hard to get to. Yeah, it was a mess. Logistically, it was just really complicated. So I think what they did is they had everybody fly to Reunion 3 days before the cruise, get tested before they flew, and they get tested once they arrived in Reunion. And then I think the rules that they said were if one person has it, nobody goes, which is insane because you spent all this money to get to the southern Indian Ocean. But luckily it went off, and so then she came back in March, so it worked. But it was chaos. It's just straight chaos trying to make it all happen. So, it was just really hard. And flying in and out was really hard. And field work has been hard. Ship schedules are hard. Everything's just really hard.
Oof, that is brutal. I mean, we were complaining about occupancy in lab, but not being able to collect samples, just, I feel for you.
Yeah, there have been a lot of people in our department who've been really messed up and had field work and just haven't, not just oceanographers, but other people who haven't been able to go out and collect samples. It's just really, really hard, especially if you're a grad student and you only have a few years to try and get these samples, and you might not have another opportunity, or it's tough. So everybody's trying to be flexible, do the best they can.
Makes sense. All right, so a little bit more lighthearted. I have 2 questions left on my list. We have 10 minutes left.
Okay. All right.
What is the Earth is flat / anti-vax equivalent in your field? Is there some kind of just irrational dogma that exists?
I can't really think of one. Maybe some of the audience has some suggestions and they can help me out.
Yeah, it's one of those, like, I don't know how much people think about, nitrogen cycles and whatnot to make up a conspiracy.
Not that much. Yeah, I think that's the
Sea
thing.
monster.
Is that we just don't have that much attention, and so like only people who really are into it.
Chelsea's answer is sea monsters, which is correct.
Monsters.
The Meg. We're back to it again.
Exactly. Full circle.
I mean, I guess that is
I hold out hope. I hold out hope.
the Bigfoot of the ocean, sea monsters. All right.
I did collect a copepod in one of my deep sea casts, like down to like 5,000 meters or something. I collected a copepod. It came back up to the surface. It didn't explode. I thought it was going to explode.
Ooh, open set has haunted pirate ships.
Yeah, I don't know about that. I haven't heard any stories about that at sea. There are definitely pirates out there. I've been in pirate lockdown before.
Close enough. All right, so I got one more. What was your I made it moment as a scientist? Because clearly you've made it. You're playing Mario Kart on Ask a Scientist Gaming. You have reached pinnacle scientist. What was your moment?
I don't know. I still feel like, am I a scientist? I don't know. I told my daughter about this. She's like, you're going to get paid to play video games? I was like, no, I'm definitely not being paid to play video games. This is what we call outreach.
Yeah. Broader impacts is the phrase. Broader impacts.
Yeah, exactly. I think probably my first sole PI NSF grant. That feels pretty validating.
That was the big one.
Yeah, pre-tenure and your community is willing to invest in you, and you're the only person on it. That's good.
Yeah. That's
feeling.
like the scientific community is willing to bet $500,000 on your science. Like that is a non-trivial investment in you as a scientist. So that's
right.
No, I agree with it. That's a non-trivial event. And every time I get a grant, I have the same feeling, right? Like who the hell? Because imposter syndrome is strong, right?
It's very strong, yeah, for reals. And I learned early on, you write a grant and then you forget about it because you just assume it's not going to be funded, and so you move on and you start working on the next one, right?
Yep.
So I do not assume that any of my grants will be funded because we have 20% funding rate.
Yeah.
So.
Well, and what's even at the 20% success rate for those of you that haven't submitted grants and haven't had to deal with this, it's like the top half of awards are hard to distinguish. Like there's a threshold where all of them are fundable, but you can only fund 30% of those. Right. And so it's like roll of the dice luck.
Yeah, literally. Yeah, at some point it's just chance, and then it's, who does your mail reviews and who does your panels. Do you guys have panels in your field? We have panels and mail reviews.
It depends what program, but yeah, we have a mix of them. And it comes down to who read it, when they read it, how they were feeling that day. Did they have a bad day? Your reviews could swing it so you don't get money. It's just... And
my impression is that during COVID, all the reviewers are grumpy. I have never seen such grumpy reviews in my life. Wow, everybody's feeling this. So everybody, and I as an editor too, right? And people are not having it.
Just the tone is really universal right now.
The tone is something. Yeah, the tone that is happening these days is really something else.
I believe it. We're in a year of this stuff, and but that's like, we want to believe science is this bastion of, objectivity, but it's people making decisions, people being about money, about funding, about, papers. It's just how people feel sometimes. It's crazy.
Yeah, I have a colleague, and they said, if you think scientists like we're just a scam and we're getting rich, or like, I don't know, we've got to. They're like, you should see how mean we are to each other and what we say to each other in the peer review process, because it is brutal.
Well, that's what's crazy to me about like conspiracy theories, like, evolution is a scam and all the scientists are in on it. But like, if I could debunk evolution, I would do it immediately, and I would collect my Nobel Prize and wave at every one of you. Like, I would wreck every scientist I could.
Exactly, right. I'm like, so that was also her point, right? Like, if you have consensus on anything, it's a done deal.
Fact.
Because it's really hard to get anybody to have consensus in this field. Like, we just destroy each other, and it's brutal. And so, people just don't have any idea
how brutal it is. Yeah, like we will spend hours debating where an electron goes. Like it's the most nuanced, trivial thing that doesn't really impact anything, but it's fundamental in nature, and we will fight to the death to get that answer right.
Oh my God, yeah. For years. And it's like, oh, Jesus. Yeah.
Yep.
There's got to be a better way to spend your time sometimes.
With that said, I love it. I like writing reviews. I love reviewing papers. I'm that guy.
So I try really hard to be constructive, right? And and I think you get better at it as you go, right? And you sort of figure out like what is a more constructive way to phrase something, and like is there an alternate way to interpret this, right? And could you interpret that signal as this, and how would you be able to differentiate, right? And you sort of try and phrase things as constructively as you can.
Man, I had a meeting with my regular group meeting with my students, but I had to share with them a series of reviews we got recently because the author of those reviews was just so actively aggressive in their commentary. Like, I can't believe the authors didn't pay attention to this nitrogen atom in this particular scheme. I like just say
nitrogen.
I think there should be a nitrogen here, rather than actively confronting us with it. It's just, I don't know. So it goes.
Yeah. People are not in a good place right now, and you can tell that in reviews.
Reese's Pieces. Final question of the night. How much of the review process is improving the writing versus critiquing the science?
I think it's like 60% luck of who gets to review your paper.
Luck or unluck? I mean, it's hopefully all constructive, right?
Mm-hmm. Yeah, and the editor is supposed to play a role, but it's just so... It doesn't seem to be very systematic. So it really just sort of depends on who reviews your stuff and when and where. So I'd like to say that it was based on more or less of anything. But
all right, so we are at 11 o'clock. Do you want to finish the final? This isn't even the final race, is it?
How many? Do I have 5 races?
I think so. 3 more, 2 more.
Okay, I'll finish the cup. Yeah.
All right.
If you can handle it, if you can handle seeing these.
We can. I'm going to run to the bathroom. I'm going to be right back. Yeah. Somebody put questions in chat.
Look over at chat. Maybe I'll actually do better this round. Maybe. Oh no, in the lake. Sad.
Sorry, I try to hold off going to the bathroom, but it's only so long after 5 beers.
Oh my God, that's impressive.
I know, this is my drinking night. We don't have Saturday or Friday nights anymore, right?
Yeah, no, we live at work now, right? That's what it is.
Ooh, what's the longest paper you've had to review?
I don't know, because you know what, we get them usually they're double spaced. How do I get rid of this hedgehog?
The bumper buttons, your left and right bumper buttons should get rid of them. Just keep smashing those.
Yeah, button mash. I'm good at the button mashing. If I launch a red shell, there we go. Okay, launch a red shell at it.
So double space Word document, longest paper.
Yeah, they just get, I don't know. They get probably 40 pages, double spaced or something like that. But then that doesn't include like the figures and tables. Like that's just the text. They're so long.
Oh,
and
you guys.
Proposals are actually longer, right?
You guys still separate figures from text?
Depends on the journal.
I see. I just did one that was 1,700 lines long. I don't know.
That's not right.
Yeah, what is that? A review article? That's horrible.
Yeah, it's not fair. Yeah, it's just hard. People are having a hard time both writing and it's just to get reviewers. It's all just hard. Everybody knows it's just hard right now. So we're all that little digger trying to get the boat out of the Suez Canal, right? And we're all just doing our best to climb it out.
We had a notable lack of Suez Canal comments / questions / memes.
Yeah. I loved those memes for the Suez Canal, but I totally thought this.
Oh, Reese's Pieces. I'm still in awe of that 500-page manuscript from the Japanese mathematician that only like 4 people in the world are trained in the same area.
That's not right. That's not right.
Most journals have a page limit or a word limit or something, because don't be that guy. Just write a book if you're going to do that.
Yeah, that's right. That's for a book. You can't do that to your peers. Nobody will tolerate that anymore.
Yeah.
Maybe they do that in math. I feel
well, there's a direct correlation. Like, people have studied this with science and nature articles between the title length and the page length in terms of number of citations. So the shorter the title and the paper, the more citations it's going to get.
Oh, good to know.
And again, that's again science lacking this objectivity. That's a subjective thing, but it matters.
It's good to know.
Yeah.
Right, I will.
Yeah, it's a National Academy of Sciences paper that basically showed the graph of the correlation between length and citations.
People are tired of reading. They're just tired.
We're all just exhausted.
Yes. Have you seen the bags under Ken's eyes?
Oh my God, it's getting bad.
It's bad. Yeah, it's tough. Although the academic semester is coming to an end, so.
And we get
a small reprieve.
Couple week break before summer starts, and so that'll be nice.
Yeah. Do you teach during the summer?
No, we can if we want to. If we want an extra paycheck from teaching, we can. But it's one of those, like, the undergrads are completely off campus for 2 weeks and then they come back.
Yeah, you can tell.
Yep.
Okay, guys, I've got the groundhog stuck to me again.
Pacific Plankton, thank you for the follow. If you have a last-minute question, Angie's rounding out her Mario Kart round, so throw them in chat right now. Again, thank you for the follow.
Oh, I got rid of the thing. I don't know if that was me or if it just got tired and it popped off.
And shout out to everyone that is following us. We recently passed 200 followers, so thank you for that. Building an audience, people that want to see mediocre gameplay and expert science. With that said, you did win the Mario Kart Gold earlier today.
I did,
in the Mario Cup.
And this is like, I just keep collecting hedgehogs. So, and, I haven't played this version of Mario Kart in like maybe 30 years.
When we do it in person, we'll have to bust out battle mode.
Yeah, totally. Battle mode. That's what we did for New Year's Eve at my house, is we did a battle mode.
Pacific Plankton, what is your favorite invasive phytoplankton?
Oh, I don't know. That's a good question. Invasive phytoplankton. I mean, phytoplankton, are they invasive? They're just sort of everywhere, right? They're kind of causing problems. Just in different abundances.
Oh, that's interesting.
I'm just going to go with Trico because it's always my answer. So I don't know if it's invasive or not, but I haven't heard of an invasive phytoplankton. Definitely other critters, but yeah, I don't know if phytoplankton are invasive.
I mean,
I'm not familiar with phytoplankton being called invasive. That doesn't mean that there aren't some. I am a geochemist, not a phytoplankton. I hang out with phytoplankton.
All right, it's 12:06. Should we call it quits there?
I think so.
Oh, last question.
Right? We didn't go to
11:06. Sorry.
Okay.
Nialis has.
Oh God, I'm not trying to start. God, make it stop.
Nialis has one more question. What is your favorite game?
That is good. It's probably Mario Kart on the Nintendo 64.
So we just, for everyone's knowledge, we tried Mario Kart on the Nintendo 64. We had a hard time streaming it remotely. So when Angela's back in person, we will be playing Mario Kart 64 as well as Smash Brothers. So join us whenever that'll happen. Probably summer or fall. We'll figure it out.
There you go.
but yeah, it's time. Angie, thank you for stopping by. It's a pleasure to
Thanks,
have you
guys.
on. A little bit of Super Mario World, a little bit of Super Mario Kart. Thank you for everyone in chat that's asking questions. You're what keeps this stream alive. So we really appreciate the follows and subs and questions. It's why we do what we do. So our next stream will be April 15th. That's Thursday, and that'll be with Stephen Leonard. He's a bio-nanotechnology person using lipid layers to try to control signaling and measurements and all sorts of really cool things. So follow us on Twitch to get the notifications. Follow us on Twitter. We'll send out a notification a week ahead of time. If you can't make the stream, check out YouTube. This video will be up about 48 hours from now because I'm restricted by Twitch to delay the posting. But yeah, if you want to see the questions and some awesome, I guess, oceanography jokes, check out the video on YouTube. But Angie, it's been a pleasure. Any parting words for the audience?
Thanks so much, you guys. Thanks to Nice Boulder and Chelsea in particular. Nice Boulder, I'm not sure exactly who you are, but I'm sure you were in one of my classes. So thank you, you guys. Send me an email. And thanks for coming to everybody. I appreciate it. And thanks for having me, Ken. This is great.
Yeah. Thanks again. Always a pleasure. We'll see you guys in a couple weeks at the next Ask a Scientist Gaming. Until then, we'll see you guys later.